Day. This is our first one of these. It's an NIH-wide event. Just a few things. Please keep your microphone muted. If you have questions, we encourage questions. Just type them in the comments section, and I'll be able to pose those to the speakers when we have the Q&A. So for our first speaker, who needs no introduction, but I'm still going to do it anyway. We have Dr. Jody Rutter. She is the acting director of NCATS at NIH. She previously was the deputy director, and now she's overseen the planning and execution of our center's complex, multifaceted programs that aim to overcome specific and operational barriers impeding the development and delivery of new treatments and other health solutions. She received her PhD from Dartmouth Medical School, and she completed a postdoc at NCI within the Division of Cancer Epidemiology and Genetics. And with that, we welcome Dr. Rutter to give us some opening remarks. Thank you, Joni.
All right, well, thank you, Jamie. I really appreciate it. Thank you for the invitation to be here today to give you some opening remarks. And I just want to also take a moment to thank you, Jamie, and Alex and Sita and Sam. I know you've been really deeply entrenched in the organization of this meeting, and it's an important one. And so I'm really glad to be here, and I can't tell you enough how much I appreciate your work going into it. I want to just start off with a bit of a confession. So we're talking about a specialized platform for innovative research exploration. And that's the acronym that we call this is ASPIRE. It's ASPIRE Day. And my confession is, and this is really for the people I think who are stuck out in the fire alarm. So they're probably not hearing this, but I've never really liked the acronym for this program that we call ASPIRE. The people who know this are in the fire drill, I think, but it's really nothing personal. I just thought it didn't really capture the essence of what we're talking about. But then there's a but. But then I started to deconstruct the word, and more as a word, less as an acronym. So Alex actually was just talking about this before the meeting started, and basically it comes from the joining of two Latin words, ad, meaning two, and spirare, meaning breathe. So over time, that got shortened to aspirare. And then even over more and more time, it got shortened to what we use today in English, which is aspire. And what it means is to direct one's hopes or ambitions towards achieving something. And in the literary sense, it means to rise high, to tower. So then I started thinking about what this program is. And in a nutshell, it's to bring together high throughput biological screening, AI, machine learning technologies, to have an integrated effort to explore biologically active chemical space. And if that works, it stands to disrupt the chemical genomics and drug discovery field in the next decade. So over the past 15 months, we've really struggled with COVID-19 pandemic, and we've been fighting. And if there's one thing that pandemic has taught us, it's that there is a critical need for rapidly developing potential therapeutic interventions for all diseases. So our mission at NCATS is to do just this. It's to turn promising research discoveries into health solutions. And our translational science strategy is twofold. Every project that we take on has to advance the science of that project, and it has to identify and address the costly and time-consuming bottlenecks that slow down or prevent us from moving that translation of the research forward. And that allows us then, if we can address those bottlenecks, we can increase the pace of testing and treatment for many diseases at a time. And our driving hope with that then is to get more treatments to more people more quickly. So everything that we do is a collaboration as well. And we have the expertise in the preclinical and the biological space, as well as the high throughput and AI ML capacity to cover the breadth of these needs. And we rely and we need partners to cover the depth of a given disease space for a needed project, so that we can make sure that we're being very specific in our activities for preclinical drug development. So we need that breadth and we need that depth. And what Aspire is today at NCATS is really a firm foundation for these efforts. We have a state-of-the-art chemistry automation facility utilizing flow and batch chemistries in modular systems and integrated platforms with informatics tools and databases to track all of the biological and the molecular space and the activities. We have combinations of mobile robotics and informatics tools to accommodate those approaches in chemistry and how the biology is being developed. We have multidisciplinary efforts between automation technologists, engineers, chemists, biologists, informaticians, and all of these groups are needed to really accelerate the preclinical discovery. And so these collaborations are absolutely essential. We're working with academic and biotech collaborations and expanding molecular diversity. MyTide Therapeutics, UMBC and Stanford, NCI Frederick, all of these groups are involved in helping us work on therapeutic peptides and protein libraries. We also have industry collaborators to expand synthetic chemistry automation for off-patent drugs, and thinking about on-demand pharmaceuticals and Lilly for new chemical space. Working with Lilly Life Sciences Studio and Stratios, L2S2, so on and so forth. There are a variety of groups who are involved in making this a reality. And I should say too, that with this, NCATS is really pushing the 3D approach of developing, demonstrating, and disseminating our models of translational science that others can follow when creating this automated research environment of their own. We're in the process of developing and demonstrating, and the next steps are really about disseminating and continuing that further work of demonstration and dissemination. Importantly, there's got to be a focus on secure and effective interconnectivity across the research workflows. This is going to allow more efficient operations by establishing a deep Aspire community that's going to set the standards and invoke that collaborative buy-in so that the results generated can be trusted by everyone. So what's next in some of that? Well, we need labs demonstrating an operational Aspire synthetic chemistry platform. And I think that we're very close to doing this. We should be able to do this in the next 6 months, 6 to 8 months. We need to demonstrate and disseminate publications, for example, and start to publish things on how we're innovating around integrated automated chemistry and biology annotations and workflows. And hopefully we can do a lot of that publication, start those publications later next year. And then we need to develop strategies for longer term sustainability for this effort. So we need to work on getting that buy-in so that we can figure out how best to get those long-term strategies in place. And to do this last bit on sustainability, it's really about describing the value add and the opportunity to contribute to drug development pipelines that's really in a kind of a dire need of an overhaul. And so we need, this is where Aspire can really come in. So the success of Aspire then is about new drug candidates. It's about novel targets. Novel targets are essential to new approaches to fighting diseases of all kinds, and modulating those novel targets will require new chemical structures and new drug candidates to be produced. So the Aspire solution combines automated chemistry, AI/ML, and biological testing, and seamlessly bridges the chemical biology space, the computational chemistry, and the pharmacology into that preclinical drug discovery. So with over about, there's more than 10,000 diseases out there. There are treatments for about 500 of them. We have a long way to go. We need new approaches to close this gap. Aspire can be that bridge. Aspire can give us what we need, and that what we need is room to breathe. That gets us back to the word Aspire. So now I'm all in on the Aspire acronym. I love it. So keep using it. I look forward to working with you to expand our targets and drug candidates so that we can accelerate our pace of getting more treatments to more people more quickly. And I want to thank every one of you for your participation, your dedication, and your aspiration to transforming drug discovery and development. And so with that, I know Alex Godfrey is going to say a lot more about the meeting itself. And so I'll turn it over to Jamie and Alex to take us to the next steps.
Thank you so much, Dr. Rutter. We really appreciate you and taking the time to speak to us. And just to kind of advance the program, I'm going to introduce our next speaker, Dr. Alex Godfrey. He's the automated chemistry consultant and essentially our project lead on Aspire. And so Alex is a PhD trained organic synthetic chemist with over 40 publications and patents, who is running our Aspire program. And prior to this, he spent over 25 years at Eli Lilly and Company, where he was the lead on developing a state-of-the-art chemical synthesis facility called the Automated Synthesis Lab, or ASL, and also early work on their 2.0 version, the L2S2. In the interest of giving Alex the most amount of time to really talk about what Aspire is, essentially, I'm going to turn over to him. And thank you so much, Alex, for talking.
Great. Thank you, Jamie. Let me get the screening situation going here. Can everyone see the slides and hear me okay?
Yeah, looks good.
All right, great. Well, okay. Well, thank you, Jamie, for the kind introduction. And thank you, Dr. Rutter, for helping us kick off this great event. And finally, thank you for joining us today at this mini workshop where we've been putting together a series of stories relating to a really cool project that NCATS has embarked on. First, let me extend my sincere appreciation to Dr. Jamie Dahl for his work putting together and coordinating this outstanding program, particularly given the fact that no sooner he'd been hired at NCATS, he was put on task to organize this event. So a Zoom applause for Jamie. Hopefully the title of this talk summarizes what we're up to. And hopefully by the end of this workshop, you'll have a better sense of what we intend to achieve this ambitious goal of achieving real-time translational science. So let me start by sharing with you the NCATS mission, while highlighting that at the very heart of the mission lies this desire to innovate, right? And since this is a workshop dedicated to Aspire project, I will also invite you to visit the NCATS website dedicated to some additional background and previous activities associated with this project. Our goal is to keep the public informed of our progress through this website, through scientific publications and other public forums, not unlike this workshop. So why Aspire Day? Well, number one, it's part of our mission. It's one of our 3Ds, right? To develop, demonstrate, and disseminate. We'll also be doing some demonstrations throughout the breakout sessions. Secondly, we wanted to share some of our early success along with some of the thinking about some of the bigger challenges we'll be facing. And thirdly, we hope to inspire you to join the effort however you can. So what makes Aspire particularly different, especially if you compare it to other ongoing efforts leveraging automation and life sciences research? Well, one clear distinction is that it's being done at the NIH, and with it comes the opportunity to create a publicly accessible platform that produces new research standards and an immersive scientific immersive scientific collaborations. It also offers the opportunity to create a more open framework that others can either copy, use, or learn from. Secondly, we are attempting to build something that can rapidly adapt to an ever-changing research landscape. The metaphor I like to use here is it's not enough to just build a bigger, faster car. We also need to build a better cockpit to better maneuver in the space that we seek to explore. I have an excerpt here from an article written by our former director, Dr. Chris Austin, where he lays out the meaning and purpose behind translational science. It's a great read, by the way, so I highly recommend it. So to quote, translational science seeks to elucidate general operative principles in order to transform translation into an empirical phenomenological process into a predictive science. So I've underlined the underlined notation is mine. For me, it points out the heart of the matter, and that is that it takes really good tools to elucidate these general principles. So to me, building better tools means getting better at answering the what if questions. At the heart of all scientific endeavor is the hypothesis testing cycle. I'm borrowing this cartoon from a review article by Gisbert Schneider to illustrate my point. Scientific inquiry can be broken down into two key activities, deduction and induction. In other words, from ideas and hunches to parameters and procedure. It's easy to see how automation connects to the inductive component, but what about the poor scientists back at the bench? What can we do to help them? Well, the reality of the matter is that in over 100 years, not much has really changed at the bench, right? Aside from slight differences in PPE and no longer being required to mouth pipette, arguably not much has changed. The truth of the matter is the bottleneck to elaborating on new ideas resides at the bench, not with the robot. So, thus we introduce Aspire, where we aim to reimagine the research laboratory ecosystem. Aspire, by the way, stands for a specialized platform for innovative research exploration. So what are we exactly proposing to do? Modernize the lab bench or build an integrated automated solution? Well, the answer is not one or the other, but both actually. So simply put, standardized methods are captured at the bench, and that may include introducing new technologies, and then pass to the robot for routine execution, thus creating a system that can actually evolve over time. So let's examine further problems that Aspire aims to help resolve. We want to increase the value of the data we collect, not just by making it more precise, but delivering it in a timely fashion as well. The more time between collecting and responding to data, the more expensive the whole venture becomes. We want to improve the confidence of the results. This means that accurate capturing the context in which the data was generated matters as well. We want to improve the ability to generate reproducible results. And this becomes an implicit expectation when we let robots do the work. We want to accelerate the dissemination of results and knowledge. And what I mean by that is that we want to be sure we're not just marking time faster in place by generating more data. We want to see measurable acceleration in milestone achievements. So how exactly do we plan to achieve this? Well, let me walk you through these three main points. We begin by laying out an informatics framework, then a functional lab model that will inform, support, and evolve the fully integrated solution. And then lastly, but really concurrently with the others, standardize everything, basically, the framework, the workflows, and then disseminate information as quickly, effectively, and as broadly as possible. Underneath each of these objectives are bullet points of various approaches currently being explored and will be further detailed in later talks by members of the team. So represented here is Aspire's closed loop automation process, the way we envision it. Beginning at a reasonably high level, we have in silico design, linked to reaction preparation, then isolation, then purification, and finally biological testing. And then the data from this is used and re-enters the iterative loop. Of course, as one dissects these steps further, as shown on the right, much more complexity in the process is revealed. A well-documented bottleneck in this process is purification, mainly because it's inherently a relatively slow serial process, and simply numbering up is a really expensive proposition at this point of the technology that's available. But wait, there's more. There are a number of additional work streams that need to align with our closed loop work stream that I have shown here with the little icon on the left. We need to define an optimal consumable strategy that, together with the data being collected from experiments, will need to be coordinated with the informatics work stream, which also requires the right automation infrastructure and monitoring control. And lastly, it's important to lay out a strategy for moving new technology in and old technology out. So how does one take on such a daunting task? Well, it's much like eating an elephant, right? You do it one bite at a time. And what I mean euphemistically here is that a deep understanding of the bench level workflows is key. It's important to know exactly what the scientist is doing at the bench, not what you think they're doing. So this leads me to the next slide. So the way we see this playing out most effectively is by laying out actually parallel work streams. One that represents an optimized digitized lab bench, and the other representing the integrated automation solution. Internally, we refer to these as Aspire 1.0 and Aspire 2.0. There's actually a lot more design development crosstalk between these two than is reflected in this diagram. It's not really moving from one to the other, as the arrow might indicate. By design, the internal working parts of these work streams look and function in strikingly similar fashion, just with more human-in-the-loop interactions associated with the NCATS dashboard. I'm not going to go into much detail here. Instead, I'm going to defer to our other team presentations, and particularly by those by Megha Verma, Vishaka Goyal, and Matthew Bijou, to see these playing out more concretely. However, the basic idea here is to create a platform that directly assists in creating new experiment templates from exploratory work that can be directly used in an integrated solution. What this effort is starting to look like is represented here. On the left, we have the NCATS dashboard, and on the right, an early design concept of the integrated Aspire facility. To digitize the lab bench, we chose to capitalize on already existing inexpensive IoT technologies to pilot this effort together with Microsoft's Azure IoT Cloud Services and their excellent suite of tools, which really expedited the development of a functioning prototype. One of the primary goals of this effort was simply to use this rapid iterative prototyping effort to collect requirements. The idea was to simply test the framework and understand that it would be possible to replace it with other types of cloud services implementations if we so chose at a later point. On top of this, we then lay an informatics framework that essentially tying these systems together, which we're calling the AICP, or Aspire Integrated Computational Platform. Again, I will defer giving a more detailed description of the AICP, and instead invite you to listen to Georgi Zaharanci-Kohalny's talk, who will be presenting on this topic a little bit later. So here's a representation of what an integrated benchtop workflow for chemistry might look like. The idea is to have work coming from one workstation, understand, if you will, what they're receiving and facilitate processing of that sample. For example, fractionation information coming from the chromatography unit would be automatically transferred to the evaporation unit, thus facilitating the pooling of fractions and greatly reducing the burden of having the user orchestrate this entire effort. Another development objective is also to ensure that users can readily switch from serial operations to batch operations as their situation calls for. The way this is playing out currently is that we are iterating quickly and repeatedly through current work streams. I know that sounds somewhat redundant, but what I mean to say is that we're attempting to multiple identical or similar experiments, and then with each iteration look at making refinements and collect key requirements that will drive the process more efficiently, which means paying attention to things like the consumables being used and the information being collected and passed along. I would add that we have found it critical to do this in close partnership with the med chemists, both to understand exactly what they're doing and to build solutions that actually work for them. One of our top level goals here is to create a situational awareness, a complete situational awareness for the scientists in the lab, either through their computer or mobile device. So switching gears here a bit, one of the key tenets of automation is standardization. You know, as a scientist and particularly as a chemist, that is, I should say, and continues to be a very hard pill to swallow. You know, setting up arbitrary constraints seems to be antithetical to a way a chemist thinks about chemistry. And after all, chemistry is already hard enough without having to add all these arbitrary constraints. So lane here is meant to represent that selected set of constraints that we select will provide the best chance for creating a clear line of sight, if you will, for the whole process, from reaction preparation to biological testing. So the vessels shown here were selected as our ideal standard reaction vessel, with features I'll enumerate here. They're commercially available, extremely sturdy, and well suited for both manual or robotic handling. The crib septum seal offers the ability to set up reactions set up and run reactions under inert conditions if needed, and allows the vessel to be incubated across a broad range of conditions, temperatures, and internal pressures, or in the case of flow, serve as a collection receptacle that then subsequently gets processed as if it had come from a batch reaction. And the range of working volumes hits a really nice sweet spot in terms of the volumes most medicinal chemists are used to working with. So the magic starts to happen when you look at when you lock down the chemistry scale to discrete molar target yields. And then when you examine the range of molarities this arrangement affords, a synthetically attractive range of reaction concentrations are revealed. It so happens that this diagonal array of values encompasses a fairly tight nominal concentration range across the various scale factors, whereby the opposite diagonal reveals a more extensive range being made available. So the cool thing about the selection of discrete reaction scales is that it offers up a whole new way of thinking about reagent delivery options and techniques as shown in this slide. ChemSpeed has recently come up with this innovative mechanism delivering precise pre-measured quantities in sealed glass ampules. These ampules can be robotically inserted into the reaction vessels, regardless of the physical properties of the chemicals contained within, and then broken open either mechanically or under a gas pressure, which in practice results in pulverizing the glass ampule and effectively controlling the precise moment the chemicals come in contact with each other, and thus offering a precise and generally useful method for reagent dispensing across a broad range of intrinsic chemical properties. So on the right are some interesting pre-formulated commercial offerings of some common reagents and catalysts, which are also quite interesting. More recently of note has been the development of these chemically coated glass and polystyrene beads developed recently by AbbVie scientists. There are a number of appealing features to this technique, namely that the materials tend to adopt the flow characteristics of the material they're bound to, thus opening up a more robust powder dispensing option. The more uniform particle distribution also makes it practical to manually dispense these using 3D printed scoops as well, calibrated 3D printed scoops as well, as AbbVie's demonstrated. Since the loading capacity can be tightly and uniformly controlled between 1 to 20 weight%, it makes it an ideal method for accurately weighing out really small quantities of material, and thus making it ideal for engaging in small-scale, high-throughput experimentation, as they demonstrated in a follow-up article. And more recently, reaction screening efforts involving biocatalysts, which I find really remarkable. So speaking of reaction screening, we're thinking reaction screening might be the way to improve our reaction performance under necessarily standardized conditions. Take this nominal example, that I've shown here, using the lower end of our standardized scale, which I described earlier. Demands on material quantity for conducting such a multifactorial screen at this lower end of the scale may still prove to be somewhat prohibitive. However, if we switch to lower orders of scale, but maintain the similar standardization approach, we could introduce interesting approach to screening for better conditions when faced with certain subset of recalcitrant substrates within a given set of synthetic targets, let's say. Albeit, some extrapolation of reaction conditions may be necessary to accommodate scale-dependent factors like mixing, heat transfer, and effects like that. The goal here, you call it a stretch goal at this point, is to have the system automatically select improved conditions directly from the screening data. Having standardized chemistry templates might actually make this, might actually help in this regard. So this led us to mapping out a potentially unique overall reaction screening strategy, one that also leverages the enterprise-level reaction analytics platform that we're currently building. Reaction screening becomes this four-pronged strategy, beginning with the building of on the top here, a direct user assistance platform through the development of standardized kits and templated procedures. Users can use the system themselves on a more user-friendly platform. The next is the next to the one to the right is a separate platform to explore chemistry areas of strategic interest as revealed by analyses of our reaction network. And a third one is a platform to profile new technologies or methodologies that appear in the literature or elsewhere. And it's actually a system we have in-house already. And Dave Calabrese will be presenting a lot of the work he's been doing on that platform. And then finally, a platform to support automated synthesis efforts. So in the context of this last screening objective, we have in the works this new platform that we're designing with ChemSpeed support. The goal here is to create a more distributed module platform to improve throughput, which leverages ChemSpeed's most recent development of a robotically controlled plate sealing mechanism shown here. So with regards to the Aspire facility, we have just recently been granted occupancy of a newly renovated space of approximately 4,400 square feet that will house the facility and supporting infrastructure. And here's a rendering of what that lab of the lab concept that we have in mind, roughly in the same perspective of the photograph I just showed you. And here's a little more detail on how we currently picture the lab being laid out. Again, we're still very early stages of this part of the project, but we're really excited to share with you what we think might be possible. So back to the Aspire loop representation. There's one very important step here that I want to highlight, and that is the isolation step, number 3 here. This is where the workup step, this is where workup occurs, essentially where the product is separated from the reaction milieu. The critical technique we plan to invoke here is SPE, which stands for solid phase extraction. To describe very briefly, this is a technique that uses solid media of varying chemical and physical properties to facilitate product isolation and resembles a fairly rapid filtration operation in many cases, together with minimal fractionation. The primary goal of SPE is to increase the probability of success of the purification step. And because SPE alone can provide some reasonable degree of product purity on its own, or at least in sufficient purity to be used in a subsequent synthetic step, it could dramatically improve the throughput of multi-step synthetic sequences, bypassing the lengthier and more complex purification step. So to this end, given the lack of any real good commercial SPE solutions out there geared towards synthetic applications, as well as certain requirements we kind of set upon ourselves, we've opted to develop our own device, and laid out here is how we envision that development progression. The engineering team here at NCATS has built the completely manual version shown here from 3D printed parts, mostly from 3D printed parts, and which is currently being used in the lab to inform the design of the more automated systems. And they've actually created a working prototype of the more automated system, and then this is informing the design, or the more product-like, if you will, fully integrated version, which is being developed in partnership with Techmatic and Automated Solutions of America. So this is, so one of the last changes I'll share with you, we've dubbed reaction templating. It's aimed at answering the question, how do we get reactions to automatically set themselves up, right? I simplistically think of this in terms of an object-oriented programming paradigm. If you define an object with the right attributes and methods, you should be able to end up with a robotic process as a program would run. Thus, features of such a system would necessarily have to be organized around a reaction ontology, afford a complete reaction annotation. We think of this as leaving no atom behind. Then a standardized representation of that reaction equation itself that is actually scale independent, and a set of machine interpretable temporal instructions. So we started this exercise by extracting NCATS ELN data, and I'll leave it to Gary to kind of fill you in on the gory details. We set out to annotate the reactions using the Next Move's Hazelnut software, and separately, we also attempted to annotate the components of the reaction. So chemists will often represent reactions like this, often leaving out key details or inconsistently, ambiguously placing components, making it difficult to assess their precise role in the reaction or even composition. So our goal is to achieve this level of annotation shown here on every reaction we work with. All that would be missing would be the temporal instruction set, which would be stored separately. So to feed our reaction templating objectives, one of the other goals is establish a set of chemistry benchmark reactions that would be developed and be used to develop and evaluate automation solutions in a more general manner. So we selected 12 reactions initially, 2 examples of each to make up about 24 total reactions. That generally represent a common set of reaction types a met chemist is likely to encounter in the lab, and also represents a reasonable diverse set of required manipulations and conditions. The goal here is to define and characterize the set of core reaction standards that would afford us a means of getting an apples-to-apples comparison of the performance of any automation setup. We also want to create a broad but consistent annotation and reaction stoichiometry scheme from which these screenshots are intended to represent. With these benchmarks in hand, we'll have a way to more accurately measure functional improvements and address automation gotchas. So we anticipate what we anticipate will happen while drilling into the characterization of this preliminary set of reaction templates is that it will help inform the development of future experiment design interface that improves the experimental design setup experience for users while also improving the quality of reaction information being generated. So pictured here is what a more standardized representation of a template might look like. So next we need to deal with the temporal instruction set. The key element that gets woven into here is the standardized reaction template fixed scale requirement, right? And what's nice about this is it could necessarily preclude having to do any manual or programmatic check of the setup to make sure it'll run on the equipment. Experiment parameters would then be passed to a mapping algorithm representing the available hardware and associated consumable routing. Key dispensing parameters would be pre-encoded in the inventory information for each chemical to be used. These instructions would be encoded in some machine-readable format yet to be determined. It happens that the recent development of frameworks that capture machine-readable protocols, like the auto protocol schema from Stratios, or the ChemPU being developed by, or the KDL within the ChemPU being developed by the Cronin Lab, gives us hope that these might be excellent open standards on which this could be implemented. So in summary, these are the components that make up a reaction template. The reactions graphical representation, the hardware graph where the hardware specs you actually have determines what scale options are actually available for execution. And finally, the temporal instructions, which would include a standardized workup setup. And in this final slide, I'm depicting how we're envisioning the reaction templating process playing out. One starts with an idea for a reaction. A search is executed to find the closest reaction variant or cluster. The new reaction components are mapped into the best fit selection. The new reactions are searched in the annotated database of known chemicals to determine the method of delivery for the reactants. Scaling factors are then applied, either default ones or user selected ones, along with the temporal instruction set associated with the reaction variant. If the instructions don't match the available hardware, a new set of parameters are recommended until a satisfactory match is achieved, and then the reaction can proceed to setup and execution. So this is my final slide. The power of the chemistry AI we're attempting to develop comes from examining the many layers of abstraction we're aiming to create. At the very top, you have this very generic description of the chemical reaction, and at the bottom, a very precise elaboration of a very specific example. So what starts to look very interesting are the middle layers, because one recognizes that that the bottom two layers speak to reaction execution within a known framework, whereby the top two open up the possibility to engage in reaction discovery. So to summarize what I've covered today in three key takeaways. One, we want to transform the laboratory bench itself to better support and grow a more scalable integrated solution. We want to hone in on standards, and we want to seek out innovations that expand operability within this necessarily more constrained parameter space. And then finally, it just remains for me to thank a terrific team that I had the pleasure to work with and have put in a tremendous amount of work despite our challenges with COVID in the last year and a half. And so with that, I thank you for your attention today, and I guess we can open it up for questions.
Sure. Thank you so much, Alex. So if there are any questions, Dr. Godfrey, please put them in the message window, and I can relay to him, just because of the sheer amount of people on the call.
Jamie, can you hear me?
Yes, I can hear you, Sam.
Hey, so this is an insider question, but Alex, really great presentation. What do you think is a realistic timeline for the SPE, given the sort of the special place it has in a bottleneck that you're very familiar with, right? Because especially that serial aspect, because this whole model you want to propose, this parallel engineering, and then we have a very serial process sort of at the heart of everything. So realistically, with the approaches that, folks are working on at NCATS with try to, get beyond that serial point, what do you think a realistic timeline that you would feel comfortable saying, to some of the folks in the call that you think at least we'll have a proposed approach that we might be able to take?
Yeah, we've been, I think COVID has offered a real challenge, particularly working with outside parties. But given that, if I had to look at my crystal ball and see things kind of lightening up a bit, and then it's not just the parties, it's also them having to work with outside parties to get some of the things built up. So, but I would say, rather confidently that within 6 months we should have a working system for the SPE. But, a benchtop model, a functioning benchtop model would be my forecast.
Looks like we have a few questions that have come in, Alex. So I guess the first one I'll pose to you is, what kind of reaction description standards has our team looked at so far?
What reaction?
Kind of reaction description standards has our team looked at so far? The other option is we could have Gergely address that in his talk as well.
Yeah, I'm not sure what they mean by reaction description. Is this in the chat? What kind of reaction description standards have you looked at so far? Oh, they're starting to pour in now.
Yeah. So we could, for the reaction description, we could maybe answer that in the chat. Think about it a little bit, and we can get to some of the other questions. Mm-hmm. Another question, is there any analytical work performed on the reaction before the SPE is attempted? Or are there any plans?
Yeah, we're definitely anticipating that. I didn't get into some of those details, but definitely analytical would be an important component. In fact, one of the technologies we're exploring, actually not exploring, we're actually moving forward with, is the integration of a lab-to-lab system. So this is a centralized pneumatic delivery system for moving tubes in these pneumatic tubes, so we can kind of sort of improve the throughput of analytical functions at the sort of facility level, as opposed to an individual lab. So that's one of the things that will definitely be integrated at this, and collecting as much, one of the things I've learned about automation is you can never have too much data coming off an automated platform to understand, how well it's operating. So yes, that definitely.
Good. Another question is, how do you determine how much of, or how do we plan to determine how much of what is going to be dispensed is the chemical versus the bead itself?
Well, I guess that's going to be a factor of the loading, the loading factor. And again, I can refer I can maybe post the references, the AbbVie's references on ChemSpeed to get a little more detail on that. But
okay,
they covered it in pretty good detail in their papers.
and then I'll a couple more questions. So another question that's posed is, would this system be adaptable to screen synthetic antibodies, in particular purification of small amounts of antibody expressed from plasmids? So not just small molecules, right? Yeah,
I mean, this could feed into sort of the biology back end in terms of how we envision that coming to fruition. But again, the idea is to build a modular system, and I could see an antibody screening module being a component that could easily be integrated. And again, the idea and the way we're thinking about, and you'll see this in some upcoming talks, is that, and particularly when it comes to biology, maybe Jamie, you can address that in your talk, but some of this integration doesn't have to be kind of lock and step. We can think of looser integrations that can still be highly functional as long as the data analysis and infrastructure supports it.
Okay, and I'll do one more question, and then anything that we don't get to, we'll address. We'll send some kind of direct messages or follow in the chat. The last question: how do you, with regard to reaction optimization purification, how do you see mass spec playing a role in this, if any?
I mean, I'd see it, I think of it fairly traditionally in the context of LC-MS, just, characterizing the purity of samples and identity to the extent we can with mass spec. One of the other things we're also going to kind of sort of, we're looking at a variety of orthogonal methods for characterization. One of them will be quantitative NMR. And actually, I would point to Colin Klein's talk, where he's going to be talking about what we've been doing in terms of using quantitative NMR to establish some of these characterization criteria.
Well, great. Looks like we've got some great questions, and whatever we didn't get to them this time, we'll try to kind of address those one-on-one or try to answer follow up with people. So for our next speaker, our keynotes, I have the great pleasure of introducing Dr. Jeff Ling. So Dr. Ling is the CEO and founder at On Demand Pharmaceuticals. He served 21 years of active duty in the U.S. Army Medical Corps and deployed several times as a critical care physician before retiring as a colonel. His inspiration for Pharmacy on Demand, or POD, was sparked by lack of critically needed medicine during a deployment in Afghanistan. And this is one story I believe he's going to be talking about today. He was the founding director of the Biological Technologies Office at DARPA and assistant director of science in President Obama's White House Office of Science and Technology Policy. He's also a professor of neurology and attending neurocritical care physician at Johns Hopkins and the Uniformed Services University of Health Sciences. He has a PhD in pharmacology from Cornell, and he graduated from medical school from Georgetown. So again, let's give a virtual applause for Dr. Ling for agreeing to give this keynote to us. Thank you, Dr. Ling. The floor is yours.
No, thank you very much. And it's good to see my friends on, hi, Joni, hi, Sitka, it's hi, all you guys. It's really a privilege to be here. And I thank you, Jamie, for extending me this invitation. I think that my time on the NCATS Advisory Council still is a highlight of my personal career as well. I spent 10 years on the NINDS Advisory Council, and I was able to spend 3 years on the Advisory Council under are all of our collective good friend, Chris Austin, and of course now Joni Rudder, who's the director. So again, thank you for letting me be a part of this wonderful program. So I'll just jump right in. And I'm going to start a share screen, if you guys don't mind. And Jamie, can you guys all see this?
You can see it. So then I think just click the center. Yep. Okay. Yep. Looks good.
Good. So I'm going to talk to you about a project that we started at DARPA, and then I have spun out to try to further advance its development, because as you know, DARPA doesn't finish anything. It starts lots of things, but does not finish anything. So that is one of the things about about working at the agency and kind of making you think a little differently about science. But be that as it may, that is a truism. So Jamie is right. This story starts appropriately, on Memorial Day, a holiday, on a mountaintop in Afghanistan. So so I actually. I'm very grateful for the U.S. Army. I must tell you guys, I joined because I was poor, like a lot of people. That's why I joined. I wish I could tell you something glorious, like I was deeply patriotic and all that kind of good stuff. But really the blunt fact of the matter was I was poor. I wanted to go to college. I wanted to go to graduate school. At the time, I had no idea I was going to go to medical school too, but I wanted to go to graduate school. And that's kind of daunting. So I needed a rich uncle. And so the only uncle I had who had the kind of money to let me go to college and graduate school was Uncle Sam. And so Uncle Sam said, okay, well, you can come on and I will pay for school. But when you're done, like most uncles who cut you a deal like that, you got to come and work on my farm when you get done. So so I dutifully tromped through school, and then I decided to go to medical school. And my good Uncle Sam said, I'll pay for that too. And next thing you know, I got done and I had to go work on his farm. And so his farm, of course, was United States Army, and it was fine. The only problem is I owe so much time to Uncle Sam that probably when I die and I come back again, I probably owe 3 or 4 more years. But be that as it may, during my career, remarkable opportunities arose. And so one of the opportunities that arose was my opportunity to pay back my uncle. And so when 9/11 hit, as you guys are all familiar with it, at that time, I was actually getting out of the Army, of all crazy things. I was actually leaving the Army in 2000 and 2001. I was actually putting my papers to separate. I took the job up at the Brigham, and my wife quit her job, which, interesting enough, was with the NIH. She was actually with the geriatrics group at that time. She was a geriatrician. She took a job at the Mass General, and then lo and behold, 9/11 hit, and I actually requested to stay in the Army, and didn't feel that it was appropriate to leave at that time. And so I stayed, and Shari actually went instead back to CMS, where she's, by the way, the Deputy Chief Medical Officer, National Chief Medical Officer, CMS. And so I went back into the Army, and the rest is history. So life has a funny way of playing out. And I don't think this would have happened, what I'm about to tell you, if I did not stay in the Army, quite frankly. And so I was deployed into my first deployment. I was deployed a few times, but my first deployment to Afghanistan was in 2003. And in the background, you can see the hospital as it was at that time, which is a bunch of tents. And the person standing with me is my good friend, Jim Eklund, who's a neurosurgeon. So we were both sent to the 452nd Combat Support Hospital. And it was there that I conceived of this notion of what you'll hear about, which is a small modular-based pharmaceutical synthesis factory, basically, a factory in a box. But you would say, well, why in heaven's name would you need such a thing? And when I was there in Afghanistan, I had the opportunity of taking care of a service member who was severely injured. He was in a gunfight, and he suffered from a number of things, but most notably, he suffered from dysautonomia. And for most of you know, dysautonomia is where your vital signs are completely unstable, heart rate, blood pressure, the whole 9 yards. And I could not put him on an air evac out of Afghanistan back to Germany. That's a 21-hour flight, given the way things are. And he would not survive the flight. So I needed bromocriptine. And bromocriptine is an old drug. We all know that. And it's like all small molecules, it is basically an organic compound. So I sat down and I was troubled by this. And I realized that, geez, if this is an organic compound, it's really only made out of 3 chemicals. carbon, hydrogen, oxygen, right? That's an organic. Now, the reality is, of course, for many drugs, you have to have a little bit of sulfur, you might need a little nitrogen, maybe a little fluoride, whatever, but you don't need the entire periodic table. You just need a handful of starting materials conceptually. So I thought to myself, geez Louise, if we had, if I had carbon, hydrogen, oxygen, and maybe, like I said, sulfur source and a fluoride source, I can make just about any small molecule that we use as medicinals. And so the idea was born that, geez, if I had a machine that could start with carbon, hydrogen, oxygen source, say a pencil, a glass of water, and an egg, and then I could basically make any kind of drug I would want. And of course, that is, that's a notional thing, but it started an idea, sketching it out. And that's what gave rise to this project. To complete the story, the US Air Force was kind enough to get a jet jockey to get a box of bromocriptine from the hospital in Landstuhl, Germany, stick it in his flight suit and fly it to us. And I was able to stabilize the young patient and send him back. And he did very well, by the way. He actually survived. And I got a nice Christmas card from his wife telling me that he had enrolled in veterinary school, so that he decided to become a veterinarian. So that was kind of a neat story. The other part of that story, of course, is that box of bromocriptine must have cost $300 million because the amount of gasoline that you had to put into a jet fighter to fly a single box of drug all the way out to Afghanistan, you can only imagine what that cost. So as you bring this whole thing back together, you would say to yourself, okay, Jeff, what you're talking about is a logistics problem, and you're solving a logistics problem. But life is great. When I came home from the war, that particular battle that I was in, I got assigned to DARPA, and I got assigned there in 2004 as a program manager. And DARPA is a magical place, truly a magical place. And Chris and Joni and I are trying to help NCATS get some of that kind of little bit of DARPA magic to mix a little bit of your NCATS magic and make NCATS an even more special place. So, I really hope to continue to work with Joni on that. But DARPA allowed me to actually pitch this idea of developing basically a 3D printer for medicinals. And that became a DARPA program. And so, and you will see that it, in fact, was successful. So I kicked off this project in about 2013. So a little bit after I came back from the war. I was busy working on a robotic arm thing. And then the challenge that I put out to the community was, can we develop a machine that will be ultimately man-portable, fit on the back of a Black Hawk helicopter or a Humvee that could basically manufacture a number of key medicinals end to end. So starting with starting materials and ending with a final formulated drug, not an API. I have never in my 30-year career as a physician prescribed an API. I've only prescribed final formulated medicines, and my soldiers need final formulated medicines. So it had to end with a final formulated medicine. And so I pushed this idea out and in typical DARPA fashion. And we got some really remarkable investigators who said that, yes, they could do such a thing. And they could do it within 4 years to build the first prototype. And so the one group that turned out to be the most successful was actually a triumvirate of these three brilliant scientists who many of you probably know. One was Klaus Jensen, who was chair at the time of chemical engineering at MIT. Alan Meyerson, another chemical engineer at MIT, and of course, Tim Jameson, a chemist, who is now the chair of chemistry at MIT. So these three MIT scientists basically came up with a plan that they could in fact build a machine that'd be the size of a household refrigerator that could actually do what I asked it to do, was to start with some basic starting materials, some precursors, not quite the egg and the pencil and the glass of water, but starting materials nevertheless. That could then be go through a continuous flow process and result in basically the active pharmaceutical ingredient, the API, and then go into a formulator that could make final formulated drug. And I wanted them to do a number of drugs that were important to the military. So these were drugs such as diphenhydramine, ibuprofen, lidocaine, and so on and so forth. And you can kind of figure it out in your own minds what these might be. So it's not the entire pharmacopoeia, but it is a list of very useful drugs that we use clinically in the kind of settings that were of the US military. And ultimately, after 2 years, they were able to build the first benchtop breadboard prototype, and they will show that they could make 7 different APIs of these classes of drugs that I just said to you. So because of the way DARPA funds things, that allowed them to get a second tranche of funding, i.e. phase 2, in which case then they built out the actual prototype, and you can see Anna in the blue lab coat standing in front of it, platform that is in fact the size of a refrigerator. Anna is only about 5 feet 2 inches tall, just to give you a perspective. Justin, who's in front of Anna, is closer to 6 foot. But you see Anna has her arms inside the machine, and this machine will now make 14 different APIs, and it will make one end-to-end. Ciprofloxacin, it will actually start with some key starting materials, work its way through the API, and end up with a final formulated tablet, 250 milligram tablets. So they demonstrated that this, in fact, could work. And that was very, very exciting. And at that point is where I had gotten out of the service by that time, and I went ahead and decided to start a company to go ahead and further develop this thing. And that's where we are today. And we'll continue the story. But the mission in my mind is always the thing that drives you forward. What do you want things to do? We always start with the back end first at DARPA. So the critical mission was that every soldier gets the right medication, always, always. So on-demand pharmaceuticals, which is what I named the companies, technology, which is what you saw, makes medicines at the point of care. And it does a number of things by being a small footprint and working with continuous flow chemistry, which I'll get more into detail in a moment. Number one is every dose quality validation. I've always worried as a practicing physician about the quality of the individual drug that I may be prescribing to my patient at that particular time and moment. I have a number of patients who will tell you in great detail how they can only take one specific manufacturer and formulation of a medication, particularly my epileptic patients. There are some who swear that they can only take depakine. They cannot take generic valproate. They cannot take any other formulations of valproate. They have to take depakine. And if in fact they don't, they will actually have a breakthrough seizure. And in fact, that has actually been shown to me clinically a number of times. So I've always wanted to be sure that every dose that I give my service member is the highest quality, and that I can guarantee it. I can actually produce documentation that every dose quality, and I'll come back to that. Second is flexibility. When you're on a mountaintop in Afghanistan, you may need any med at any time. And you'll say, how could that be? I mean, soldiers don't show up, they're pretty healthy, they don't show up when they get trauma, so on and so forth. I will tell you guys that 80 to 90% of the patients that I took care of, both in Iraq and Afghanistan, 90% were civilians. Only 10 to 20% were actually service members, which I found remarkable, but it's a good thing, actually. If you actually think about it, you want your medical providers taking care of the local populace. It's one of the great things. When some of the places I've been in Afghanistan, as soon as the locals found out that the American healthcare providers were on site, they would line up outside the gate. And I'm not joking. They would line up outside the gate. In fact, I had one commander, base commander, who in fact insisted that we would go out and walk along the line, they call it, and tend to these people who would line up. And most of them needed very simple things, like some antibiotics, perhaps, maybe some analgesics, vaccinations, that sort of thing. But there would literally be about 100 people lined up outside the gate every doggone morning, and our family practice doctor with 2 medics would literally march down that line taking care of patients. So you would literally need any med at any time. Resilience. This machine has got to be resilient. It's got to provide any med under any condition, including potentially under fire, because I wanted this machine to go where the soldiers go. I wanted to go to combat support hospitals, forward operating bases, combat outposts if necessary. It needs to go where the soldiers go. It needs to be robust. It needs to be able to tolerate being on the back of a helicopter or inside of a Humvee or on a ship, for example, or just about anywhere else you guys can think of. One thing that came to our attention was source materials. And in fact, we find out that, and I'll come back to this also, that many of the source materials that you use to make medicinals actually are overseas based. In fact, very little is actually US sourced. This is not good for the United States military. The US military must be as dependent upon itself as possible. It cannot be dependent upon a potential peer adversary for starting materials for things like medicinals or anything else for that matter. And this has shown itself to be particularly important in this COVID crisis. Alex talked very eloquently about some of the challenges that are being faced during this COVID crisis, and sourcing of raw materials is actually one of them. Finally, cost. We got to make it cheaper. We got to make it better. It is really simple as that. If we want this to sustain itself, we need it to make it cheaper and we need to make it better. And I'm happy to say is that for the medicinals that this technology uses, we can actually cut the wholesale cost of drugs by 50% or more in many cases, 50%. So that's a good thing. This is a photo on the right-hand side of what the pharmacy on demand, or POD we like to call it, the pharmacy on demand platform looks like. It's been reduced down from the size of a household refrigerator to the size that you see right there. It's not, I want to be clear here, it's still a prototype. So when you open it up, there's some dead space and all that. We believe that when we get rid of all the dead space and such things, it'll be the size of a dormitory refrigerator. And that is, that what you see there is the synthesis and purification units. The formulator is about the size of one of the modulators. And we have what they call an American-made precursor on demand, or an AMPOD unit, which is the size of one of those as well. So if you put them all together, you can actually start with just US source material, work your way all the way through the process and come up with final formulated drug in a fairly small footprint, clearly, that could then fit in the spaces of which I had talked about. So when you come back to it, what is POD? POD is really a super small manufacturing unit. It's a GMP in a box. It's a good manufacturing protocol machine in a box. And the way it works is, and I'll show you some pictures in a moment, it works with proprietary microreactors and drying, filling, the whole 9 yards. We've shrunken it down as much as possible in a proprietary way. We actually currently have 67 patents on this entire process, and we're adding more daily. We probably will be up to 100 by Christmas. And it works with continuous flow synthesis. That's what it does. It doesn't work with batch, it does with continual flow synthesis. By doing all of this, the biggest advantage that we have is flexibility. We literally can reconfigure the machine to go from one drug type to another in just a few hours. In a very short time, with overlap of unit processes, we'll be able to start to even more make this rapid, so that we can switch from one type of API to another in minutes. So right now, we can actually switch between a couple of our medicinals in about 90 minutes. And we've put an advanced inline processing monitoring system that does exactly what I wanted it to do, to provide every dose monitoring. This is important because this is a completely automated system. Alex spoke eloquently again of the importance of automation and robotics. This is meant to be a push button operation. It's meant to be used by soldiers in the field. So that means not a compounding pharmacist or a medicinal chemist, but rather, or a chemical engineer, but rather an 18-year-old who's gotten about maybe 8 weeks of training, of which 2 of it will be how to work this machine. So it really literally has to be a closed system push button operation. And then we're really well on our way to that. The machines that you saw Brendan pushing, those little white boxes, actually is already at a single operator level where you can push a button and the entire process goes from end to end. And this becomes really relevant to make it competitive on the world's scene, but it makes it possible to go to the places that I really want it to go. So all those things are already built into the system. Since it's impossible to identify what type and amount of drug is needed by each soldier and each unit, we have to be able to have a good sushi list of the number of things that we can do. And we have to be able to produce whatever drug is needed in exact quantities. And the reason for that is we get rid of the cold chain that way. We also eliminate the issue of expiration dates. We don't want expiration dates. So if we only have this thing with having raw and fine chemicals inside of it, by and large, we can avoid the issues of expiration. So let's talk about timeline. Is ODP actually delivering on this mission? And the answer is yes. If you actually look at the program from its inception, it is on budget, on time. The initial DARPA program delivered the version 1 platform in 4 years. In that 4 years, it built that machine that you saw Anna's arms inside of within a 4-year period. That machine will make 14 different APIs from different drug classes. It will do a final formulation of tablets, and it demonstrated over a 3-week continuous run that it could switch in and out of 5 different drugs with only a couple of hours in between drug transfers over. So that one little refrigerator box could actually produce thousands of doses of drugs in a very short period of time. Since then, we've actually continued to refine the machine, turn it into that modular system. But what happened is COVID struck, and you guys know all about that, of course. But when that happened, DARPA turned to us and said, geez, can you turn these machines into making drugs only starting with American-made precursors? In other words, American-made starting materials. And this was a big deal, actually, because you don't want to be dependent upon potentially a peer adversary, of which China is, or potentially a country that's having its own supply issues, such as India right now. So the idea was, can we go back to things that can be sourced in the United States? And that is actually getting to be kind of hard. And there's a number of reasons for that, but you guys are NIH, you know this. And so what happened is that in addition, they also wanted us to pivot and start to make 3 new medications that we had not made before. We had hoped to be able to make them, but we were never asked to make them. And so HHS and DARPA, of course, you make propofol, end-to-end, and saccharinium, which is Nimbex. And in the last 9 months, we're working very, very hard, and we have actually shown that we can actually make midazolam end-to-end in the United States, in our Rockville office, propofol, as well as cisatracurium. We are doing so well that, in fact, that we plan to submit our ANDA, our abbreviated new drug application, to FDA by Q4 of 2021, shortly followed thereafter by about 5 more over the next 6 months. So this is actually maturing enough to the point that we can actually submit formal applications for approval to our colleagues at the Food and Drug Administration. So this gives you an idea of the proof of concept was kicked off in 2011. 2013, we actually kicked off the program itself. They marched rapidly to 2017 to the refrigerator that you saw. Over the next couple of years, it actually morphed into this. But then over the last year, we've gone up to now 16 American-made precursors. So that was all done in the last 9 months, starting with US starting materials and then making the precursors necessary to make certain drugs. We're up to 21 APIs. I learned this morning that the guys have added another one. We can now make tablets. We can make sterile injectables. And we've demonstrated already that we can make 5 different APIs end-to-end, as I talked to you about. So how does this happen, you would say? I mean, where is the magic? The magic, I will tell you, is the way I envisioned the program to begin with. And the magic is this. Is that when you think about the problem, you can actually drill it down to, it's a chemistry problem and a chemical engineering problem. This is very DARPA. DARPA always tries to break it down to what are the core task areas. And there are really just 2 task areas in this particular build. One is a chemical engineering one, which is obviously building the types of devices necessary to go inside the machine to accommodate the chemistry. And the second part, of course, and very, very critical, is the organic chemistry. Is can you, in fact, work with earlier starting materials and go ahead and produce the end state medicinals with the reaction efficiency that you would like on a small deployed distributed platform, number one. Can you take advantage of high pressure and high temperature to, in fact, speed rates of reaction? And can you go ahead and use common starting materials to make many different things? Now, we all know that paper chemistry and actual chemistry are two different things, but in fact, therein lies the challenge. It is a difficult process, and one would say an incredibly complicated process, but it's not an impossible process, and that's really an important point. So what you see is that what we have done is we've developed, and our friends at MIT started all this. I want to be clear on this. I want to give credit where credit is due. So thank you to Klaus, Tim, and to Alan for starting all of this. And I would like to say that we have built on their brilliance. And so you always stand on the shoulders of giants, and those were really tremendously giant people. But the reactors, which you see in the top box over there, is actually a microreactor. It it's a flow-through system. That's an in and out system that will run at very high pressures and temperatures, as well as having good flow rates, and acts as a microreactor. And that is how we, in fact, are able to do this under continuous flow, starting earlier in the synthetic processes. And by starting earlier in the synthetic processes, we, in fact, are able to reduce costs because we're starting with more simple, basic starting materials, commodity chemicals when we can do it. That's number one. But number two is, which is really critical, is we're able to add more steps in. And by adding more steps in, you worry about the efficiencies of these reactions, of course. And so we had to make sure that we were able to increase the efficiency of the reactions to do that. We built a number of heating elements and some other things, and on it goes. One of the beauties that we have is we created our own machine shop. So we have our own prototyping that allows us to do turnaround times of prototypes within a single day. This would be very difficult to do, especially during COVID, if you're working with outside vendors. So we even have our own, CNC machine, actually, and our own 3D printers, as you would imagine, to go ahead and do this. We actually have a pretty nice little prototyping lab for a company our size. And you'll say, well, how much drug can actually be made in this itty bitty box, talking about what you're talking about? You'll be shocked. You can actually make quite a bit, depending on the drug, especially high potency drugs. A single pod can make a whole lot of drug. So a single pod unit can make 40 million doses of fentanyl a day. A single pod can make 2 million doses of Versed a day. And I can go down this list, but you get the idea. And in fact, we found out that one single pod, one little box, can make the nation's supply of cisatracurium, or Nimbex, per day. These are all very relevant amounts. The goal, of course, is not for a single pod to make the nation's supply of cisatracurium. That's not the point. The point is that if, in fact, you can do that, that means that you only need 15 minutes to make your hospital's supply of cisatracurium that day. Then you can pivot it and have it make your fentanyl. You can pivot it and have it make your anti-infectives. You can pivot it and have it make your, antiarrhythmics, and on it goes. The whole idea is, by having a high throughput like this, you use one machine to only make as much as you need per day. So you're not warehousing, you're not having expiration issues and the like. Furthermore, a distributed system like this, say one in every hospital, allows every community to respond as they need. This is a big deal. You can imagine what New York City's pressure was at the beginning of COVID-19. It was very different than Chicago's pressure, and which was very different than, say, a local medical clinic's pressure. Local medical clinic may not be dealing with ventilated patients, wouldn't need a cisatracurium, wouldn't need propofol, wouldn't need fentanyl, but may need a lot of outpatient medicines that they're not getting because their suppliers are so busy trying to manage getting supplies to New York City who do need the cisatracurium, who do need the Versed, who do need the propofol. And so you can get the idea of what a distributed system would allow a complex system such as the United States healthcare system and meeting all of its needs and demands. So you can see in your head why such a box would be very useful to the United States military. But then you say to yourself, well, Jeff, that's all it is? Is that only good for the military? The answer is no. One of the things that we all know is that there's dual use, right? I mean, a lot of the things the US military develops, and in fact has civilian application. On your own cell phone, for example, this is an iPhone that I have in my hand here. 90% of the components that are from that iPhone, by the way, you should know, came out of DARPA programs built for the United States military. You know the thing that makes your screen turn? That was actually built for pilots, flying and doing their maneuvers to keep their screen to be oriented the way they're oriented. And of course, it found its way into an iPhone. Siri, by the way, was a DARPA program so that pilots could use simple talking to command to get the plane to fly. So these are all things that have dual use. And I believe that pod is such an example. One thing that has been common in the United States is, in fact, chronic drug shortage. This is a chart that you should take a look at. It started back in 2014, but actually goes way before that. But consistently from 2014 all the way up to COVID, of course, the number went up dramatically. But between 2014 and 2020, look at this data. These are the data of the number of different drugs that were on the national shortage list. And it always ranges between the 170s to the 300s. So there's always at least a couple of hundred drugs that are actually shortage in the United States. Saline is a shortage in the United States, as an example, for a period of time. Heparin was a shortage in the United States for a period of time. Now, heparin, of course, is a large molecule. I can't make that. But there was a number of others. Zantac was. And so we go down the list, but there's always drug shortages. And you used to say, why are there constantly drug shortages? Well, COVID-19 showed you why. It's because we're highly dependent upon overseas manufacturers for many, many things. And in fact, as you recall, the President Trump at the time wanted the US to start making active pharmaceutical ingredients, APIs here in the United States, to reduce the dependency on India and China for those APIs. However, what he did not appreciate was they got you coming and going, because many of the starting materials used to make the APIs that we currently are familiar with are made both in India and China. So you may think you're starting to address a vulnerability at one step, but not recognizing that further down, you still have the same vulnerability. COVID-19 clearly showed that loud and clear. The other thing too is that when we talk about shortages, we don't think about shortages here in the DC metropolitan area, but if you live in the middle third of the country, this is a big deal. In the middle third of the country, there is what they call a pharmacy desert. That in fact, it is many, many, many miles to get to a pharmacy and to adequate drug supplies. And it's because of strictly economics. Really, it's economics. And you would say that, oh my gosh, I mean, if I lived in the middle of, let's say, Montana or Wyoming, take a look at Wyoming, that the reason why you're there is because the population is a little dispersed. That's obvious, number one. But every clinic that's out there who are servicing, look at the VA. The VA actually has clinics in these areas. They don't have adequate pharmacy supplies, largely because of how do you keep that supply up in a cost-efficient manner, right? Second is the quality issue, of which I talked about. POD solves the quality issue. And you would say, does quality really matter? I mean, FDA approves things and it's made. The answer is a big deal. If you actually look over the last 3 years, 1,500 FDA recalls and market withdrawals were all due to safety concerns. In fact, in 2019, 15% of the custom seizures, total seizures, were pharmaceuticals. And in 2018, 7%. That's a lot. I mean, customs looks at a lot of things. And so this book by Catherine Eben, and Catherine Eben's written for the New York Times talking about this, you wonder how much of a patient's clinical deterioration is due to poor quality drug being taken. And when I would talk about these custom seizures, it isn't that the whole container full comes through as tainted drug. What happens when they reach into the middle, they find that it is substandard. And so that's what happens. Now, is it done on purpose? Counterfeit drugging is always a big problem, true, right? But the other thing too is the batch processing also is just a sampling of the quality of a batch of drugs. When you sample and make a small sample of a batch, you make the assumption that the entire batch is still good, and that may or may not be true. Right? Now, one other thing is it's just not to medicinals, it's to many other things as well. So at Johns Hopkins, my home institution, the supply guy will tell you a nice story where he bought an entire shipment of N95 masks and found out that the masks, when they tested the masks, that the outside, the front end of the container, they were fine. But once he got just one layer back, they were all bad. I mean, all bad. He had actually sent them to the applied physics lab to be tested, and they had to discard the entire $500,000 worth of N95 masks that they had bought from a reputable distributor that were not what they were purported to be. They looked like N95s, but they weren't. So it gives you an idea of that what we're used to here in the United States is not true for the rest of the world. So how in fact do we do this quality thing that you talk about? So what we do is we put a lot of online monitoring, right? We want to be sure that what we're making at each step is in fact what we want it to be. Because if it's not, of course, then we're going to end up with a poor product. So when we have online monitoring, we online monitor many things. We online monitor mass, pH, temperature, on and on. But a couple of things that we put online is Raman and infrared spectroscopy. And here are the things that you guys are very familiar with. And what we did was we wanted to be sure that for each run that we are getting what we want it to be. So we'll actually purposely perturb the system. We may change the temperature. We may change the pH. We may actually change the concentration. We will perturb it at different steps and measure how, in fact, that changes so that we actually can develop parametric models that show us what happens when things are out of sync. This allows us to be sure that the quality that we're doing is measured at multiple steps throughout the reaction so that the process, so that we can divert to waste something that isn't proper. That's number one. And number two, very critically, is if we're going to distribute these machines out, we want to be sure it's not subverted for something else. So if something is out of spec, it doesn't matter how much out of spec, it's out of spec, and that the machine itself will in fact divert to waste or just shut down completely. So this allows us to have this each dose, every dose monitoring, and more critically, it documents it. It actually, even on something simple like LabVIEW, which we're using now, it will show you exactly how good each of the process steps are. And this allows really real-time quality metrics. This is the kind of thing I think that should be allowed to the FDA. I think the FDA should have access to this. In fact, I've talked to FDA officers. Commissioner Hahn himself came and visited us, and we actually talked about how we can work with the FDA so that they can have access to this kind of data so they can have full transparency on how drugs are made. No more of this hiding behind, some sort of shielded type of processing, but really have full transparency. And in fact, for Cipro, just to give you a flavor of it, we actually monitor 29 different steps, four in the PAT process, 5 at the tablet device, and 20 are embedded throughout the entire system throughout. So there are 29 different steps are measured while we make our Cipro. But the US Pharmacopoeia also needs to work with us. So we are trying to work with USP now, right? And the idea is that what we want to do is have a very clear and efficient process that we can do this. None of this stuff that I'm talking about is new science. It's old science. I mean, you guys know this better than anybody. Why isn't it being put into place now to protect the US population? Well, that's a good question. We should, in fact, not only suggest that it should be done, we should demand that it's done. The drugs that are given to your children, to your grandparents, to your own parents, to yourselves, should be of the highest guaranteed quality. And since the technology exists for every pharmaceutical manufacturer to provide every dose quality data, then they should. Then they should. It's as simple as that. Then they should. We should demand it as a people, not that we trust you that it's going to be good, but trust and verify. The overseas dependency is an issue for the US military. It has become a little bit of an issue because of COVID-19 for civilians, but it's a big deal for the US military. And if you actually look, every generic is dependent at some step on an overseas manufacturer. At some step. If you just look at API manufacturing, just look at that simple step, 75% of it's done overseas, 75%. If you look at what an API manufacturer uses as starting materials, you'll find out that 100% of it, at some point, they're going to need something from somebody else. And so there is that high level of vulnerability. In fact, all drug ingredients, from raw material to APIs, all drug materials, ingredients, someplace along the chain, there is something that is made overseas. When you actually look at the anti-infectives, just look at that single drug class, 97% of them come from China, 97%, and on it goes. Here's why. Take a look at this. This is the standard batch processing for making APIs for Cipro. And take a look at the flags, and you'll see where you source each one of these starting materials. Nevarapine is another one. Midazolam, one that we had just talked about. And you say to yourself, why is that? I mean, Dow Chemicals at one time, DuPont, Mallinckrodt, these were, global dominating leaders. They're not anymore. In fact, the largest chemical company in the world is Chinese, as you know, it's Sino Chemicals. Why is that? And take a look at what's on the right-hand side. That is the wage index. If the US, which is the big bar, is 100, Take a look at where India and China is. It's 8%. This, by the way, is data from Janet Woodcock at the FDA, the acting FDA director. The wages to make drugs is a full order of magnitude less in these other countries. So that's another main driver that drove this out of the United States. Why DuPont and Mallinckrodt, these guys no longer dominate. They had to offshore it. That's not the only one. It's also, of course, EPA regulations, which are totally appropriate. We all would celebrate the EPA regulation. But quite frankly, the other countries don't have the same stringent requirements the United States does. And when you're making chemicals, it's a dirty business. So by using non-green technology, which is, of course, by the way, 18th century, 19th century type of technology, by using non-green technology, it is cheap, it is, and especially when you have low wages, and here you go, this is what it is. So if the US wants to recapture this market, it can either repeal child labor laws and start hiring children working 15 cents an hour so that big factories can work at a competitive rate as this, and it has got to completely abrogate all of the type of green technology mentality that we have today, and neither of which are very tenable types of solutions. So what we have to do is think smarter. We need a 21st century solution for these 21st century problems. We have to think about what to do about the wage disparity and what to do about the dirty processing. And that is, again, chemistry and engineering. Chemistry and engineering. So the other thing you want to put into place are also good quality systems. You can build these into these systems that, of which I'm talking about. We're doing it ahead of time. So we're building in a pharmacy quality system. We're also building in a framework so that the device itself will be a freestanding GMP in a box. That's key. If we want to have a distributed system and go out with my soldiers, but at the same time go to a CVS or go out to a local rural or tribal communities, a clinic, it actually has to be a GMP in a box. And we're doing all of those things. By having this distributed system, you actually solve the supply chain vulnerability that which we talked about. Think about the meat packing industry. You just heard that hackers took down the largest beef processing plant in our country, right? And it will be felt. This will be down only for a few days, but the price will be felt at these supermarkets for all of us. During COVID, remember, the pork processing industry was shut down because of COVID. And that is what happens when you have, and you guys remember the price of pork chops. So the point here is that if you have a single manufacturing site, you have a single site of vulnerability, and you want to avoid that. A distributed network, by definition, becomes a resilient network. And so why do I worry so much about it? India is having tremendous problems right now. We all know this, but its consequences are significant. When you have all of their people right now back in lockdown because of COVID-19 outbreak, that is a tragic humanitarian situation for them. But what are they not doing? They're not at work. They're not making the raw materials that we need for just about everything, not just medicines, but just about everything. So this is going to be felt downstream in about 3 months. When the current stockpiles dwindle, this will be felt, and it will have economical and it will have practical significance. Second, the one up below is the trouble that's going on in the South China Seas. So that is the Teddy Roosevelt Carrier Strike Group, which is now on station in the South China Seas. This is no little thing. When you have loss of the support of a peer adversary nation, you're going to have a lot of downstream consequences. But more to that, all of the trade that goes through the South China Seas is also going to be disrupted. So these are real issues that will have consequence. You may not care what goes on in the South China Seas, but you should be. And you may not care think that India's just going back into lockdown. That's cool, don't worry about it, but you should be, because we're all interrelated. And that's what happens when we lose our ability to actually have some self-sufficiency. Finally, POD does solve runway costs. The thing that is great is that that when you have a system like ours and you control your own manufacturing, you do control your own costs. Look what happens when you don't. I mean, these are egregious examples, for example. So on the left, we all recognize Martin Shkreli. I mean, all of us want to take him, punch him in the face. Look at that smug. You want to kick him in the rear end. But the reality is that he did something remarkable. He took Daraprim, which is as you guys know, is a medicinal for toxoplasmosis, a disease felt by HIV patients as well as cancer patients. And pyrimethamine, which is what daraprim is $13.50 in 2015 when Turin, his company, bought it and then jacked the price to $750 a pill. $750 a pill. Not the most expensive drug, I might add, but I mean, there was no reason to jump that price other than the fact is that he could and make money on it. There was a hue and a cry. As you recall, Senator Clinton, Hillary Clinton, had a hearing, and there go all sorts of bad things going to happen to this guy. If you take a look right now, go online yourselves and look at the wholesale price of daraprim, and it's $750 a pill. They didn't do a thing to it. And his response was, I only answer the shareholders. And he has a point. That it's not illegal what he did. He has a point. What's not told in this story is that $13.50. That's egregious. We all agree. But Valiant, who bought daraprim from its previous owner, I can't remember who, jumped the price when they bought the drug from $1 to $13.50. That never made the news. But I just want to be clear here. This is what happens when you only have a single supplier of a drug that is important. EpiPens, which all of us are familiar with this person who was the CEO of Mylar, and she jumped the price sixfold from $100 a pack, which is holding her hand, to $600 a pack. She talks about rebates and all that kind of stuff, but that's a lot of bullshit. At the end of the day, they're still charging $600. They jumped the price by sixfold. Why? Because they could. Guess what? Let's pretend that you had a machine in your hospital that could make epinephrine, or you can make pyrimethamine, which you very well could because they're both small molecules. And let's pretend that Jamie had a hospital also with his own machine, and Alex had his own hospital with the machine. So if any one of them, and I had one, let's pretend I was egregious and I jumped the price of EpiPen from $100 to $600, Jamie may say, wow, you could, geez, that's pretty good, but why buy Jeff's? I'll just jump it to $6.05. And Alex will say, well, forget you guys, I'll just charge $600, and then I'll come back and say, well, if you're going to charge $600, I can still charge $590 and still make a lot of money. And you see what it does. It's like a Southwest Airlines race. And instead of how high can you go, it's a question of how low you can tolerate and make money. And that's the point. The point is, if we have a distributed system wherein everybody has a machine, these two knuckleheads can't do what they did. They cannot do what they did and get away with it. So we all know the rising cost of generics meds is a leading driver. We know that critical shortages. We know about orphan drugs, by the way, is a problem. You make your own on site. You don't worry about how much you need to make. Generics are made overseas, blah, 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 blah. All these things can be managed very simply by having your ability to make the drugs yourself. So in my mind that's all great, but why do you look at me and go, Jeff, you spent 21 years in the Army, you look old and ugly and beat up. Why are you doing this? I mean, why are you doing this? And it's real simple. I'm an old guy right now. That's pretty obvious. I'm an old, broken down guy. But you know what? Before I leave, I want to do something really, really cool. And this is what is really cool. I want to service my service members. I devoted my entire adult career life to taking care of my service members. And that's the upper left-hand corner. And that's how I envision what pod will look like one day. That's my vision of it, all right? It's going to be the size of a dorm refrigerator, do all the things I just told you. But it can do all the other things, affordability for our seniors, it can help our hospitals maintain supplies, all the good things. But what I want it to go in the upper right-hand corner. I want it to go where medicines aren't. I don't want the people in the developing world to be dependent upon gifts from pharmaceutical manufacturers, because that's what it is right now. They get gifted drugs, right? Some are expired, many are not, but they're dependent upon the gifts to get it. The heck with that. When I was in Afghanistan, one of the coolest things I remember were the PRTs. Those are provincial reconstruction teams. And I knew one of the commanders of the PRTs. He was a great colonel, good guy. And he had this really interesting thing. He goes, their job was to go around to the different villages and give the villages what they wanted. And all the villages wanted pretty much what you and I would want if we lived in those places. They wanted clean water, they wanted education, and they wanted good health for their families. If they could get that, that would be a big deal. So the PRTs would dig these deep wells. There's plenty of surface water around in Afghanistan. If you haven't been there, you'll see it. But a lot of it's contaminated, all right, over years and years and years of mismanagement. So, but Afghanistan has a lot of deep water. And so the PRTs would go in and drill a 400, 500-foot depth well and provide clean, fresh water. You guys know what that means. You know, we talk about all the things we want, but if you go to many parts of the world, the only thing they would want is clean water, because clean water means so much what they can do with it. And it could dramatically improve health. And you guys all know that. I offer that, in fact, if I could put a solar panel on my machine, all my machines that you saw were small. They run off a wall, 110 volt electricity. They had to run off of a military generator. So put a solar panel on it, put a filter on the water supply, and then put it out in this place so that these people make the medicines they need, when they need it, how they need it. In other words, what you've done now is you've put the hands of their healthcare into their own. You're no longer giving them a fish, you're teaching them to fish. That to me is real liberation and real empowerment, is when you are doing for yourself. That is my dream. If I can get this machine to them, then we'll have done a great thing. And it will be another gift of America to the world. I think that's a good thing. And healthcare disparity, these all go away. Put this in inner city Chicago. Put this in tribal clinic in the middle of Oklahoma. Blah, 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 blah. And what you see is you break down barriers by doing this. You totally break down barriers, and you put in the hands of these people the ability to control their own supply. 2 billion people lack access to essential medicines in this world. 2 billion. Does that make any sense to any of you at all? It makes no sense to me. And in fact, if you go to Cote d'Ivoire, go to Uganda, go to South Africa, and there's one thing that has clearly happened over the last 5 years. The cost of HIV medications has tripled. They haven't changed. The drugs themselves have not changed. Heart therapy has not changed, but the cost of the medicines tripled. And you would say, why is that? And we all know the reason. There's only one reason why that is, and that needs to go away. So I listed all this stuff. I'm not going to go over it. You guys get it. This is on the right-hand side. You can see it. That's what Pod looks like today. I'm very proud of it. Just below it, that's a CNC machine that we make a lot of our own prototyping with, and we do that so that we can stay on a very fast, aggressive pace. And this is the build out of our new GMP suite so that we can do all the things that FDA required, full transparency with the FDA, so we can then ultimately create a GMP in a box. So in my mind, my last slide, I said with Pod, ODP is about building US capability, and it's about solving problems and solving them now. So all this program was done in a very short period of time, and most of what was done that you see that I've been talking about was done in the last 9 months. And what you want to do is all of us want to work at a DARPA fast, DARPA hard, DARPA quick pace. So I thank you for this time.
Wow, thank you so much, Dr. Ling. That's fantastic. We're having some questions come in. I'm just going to jump in though. So you mentioned the DARPA magic. What is the DARPA magic so we can copy it?
So DARPA magic is really quite simple. I've been asked that a lot lately, as you can imagine, because if you want to know who started this HARPA idea or ARPA-H idea, that was actually me back 4 years ago. I was trying to get the Trump administration to create an ARPA-H. And I got pretty far, by the way. I actually made it in Vice President Pence's office and into Ivanka's office, but I couldn't get past Jared. So I pivoted at that time and I pitched the idea to then candidate Biden. And so, as you know, this is a big deal, and hopefully NCATS will have a big part of it. I'm hoping it has a big part of it. But DARPA is this. It begins with this. It's not doing science for science sake. In fact, you guys at NCATS, and I know you guys well, trust me, I know you well. Joni will vouch that I was at all the meetings. That in fact, I know you guys well. What's different about NCATS versus the rest of NIH, what's different about DARPA and the rest of NIH is that the projects are to build capability. They're not to do science for science sake. Science is a means to an end, and the end is a capability. And what do I mean by capability? A capability means that it is addressing problems. You are problem solvers, all right? Yes, you are scientists, you're technologists, but you're using science and technology not to just expand the knowledge base, but rather to take the knowledge base and create a capability. All DARPA projects are to develop a capability. The classic example I use is the invisible airplane. DARPA set out to build an invisible airplane. Why? Because the capability was air dominance. If you have an invisible airplane, you automatically create air dominance. That was the capability. And so building the invisible airplane wasn't about finding ways to make the airplane invisible for its own sake, but was to solve a critical problem. Then people say that, do we have such a thing, Jeff? That sounds crazy. DARPA went and did it. Absolutely. The F-22 is a total invisible airplane. Make no damn mistake about it. Is it invisible when you look at it? No, but who cares if it's visible when you look at it? If you looked in the sky and you saw a jet fighter and you're the enemy, forget it, it's too late. They're about to drop a lot of fire and steel on top of your head. You want it to be invisible 300 miles away when it's coming on you. In other words, you want it to be invisible to radar. And that's what they mean by invisible airplane. That's a capability. You and I can think of many capabilities that we would love to solve in the healthcare industry right now. Many capabilities. That's where it starts. DARPA doesn't do science for science sake, it does capabilities. That's number one. The second one is having an attitude to say yes. At DARPA, when I was the director of the BTO and I founded it, I would come back and I say, everybody, I want you to have as a fallback, the answer is yes. Saying no is easy. Every jerk off in the world can say no, because no means you don't have to do anything. I said no, I don't have to pay for it. I said no, I don't have to do it. I said no, I don't have to get off my chair. When you say yes, now you have to work. And so you should have a fallback. Those who are truly innovative, fallback should be saying yes. The question you should be asking is, if it works, would it be transformative? Would it be cool? I mean, come on, an invisible jet fighter, that's cool. I want one, you want one, right? So is it cool? If the answer is yes, it's cool, find a way to say yes. But then hold accountability. That's the third element. Accountability, accountability, accountability. DARPA will fire performers. I have terminated entire programs right in midstream because they weren't performing. You have to be able to hold people accountable. I held my PMs to accountable because if they weren't keeping an eye on their programs, they were out of there. So they kept their eye on their programs. The performers have to be held accountable. That's the US taxpayer money. How many grant applications, Jamie, have you seen that said that the investigator is going to put in 10% time? Blah, 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 blah. And how much time are they going to put in 10% time, 15% time? Come on, you guys see it all the time. If you put in a DARPA program proposal and you say you're going to put in 10% time, you're automatically disqualified. You better be putting in 110% time. If you want a $20 million program, you better be coming to work every damn day. You should be coming to work on Saturdays and Sundays. You should be coming to my house and washing my car. That's $20 million. $20 million, you should be working your butt off for $20 million. That's number one. So accountability, accountability, accountability. Second, if you're going to cure, you have a cure. God bless you. You've got a cure for COVID-19. You actually have an oral therapy that will cure COVID-19. I would fund that person and then I'll hold their feet to the fire. I'd say, well, where is it? DARPA, we called our performers every single week to see how they were doing. Every week, you're going to get a phone call from your DARPA program manager. Every week. Every month, you expect a visit. Every month, you expect a visit. Every quarter, you expect to have a review. You say, that's a lot of burden on these investigators. No, that's a lot of oversight of taxpayer dollars for people who said they can do the job. That's DARPA fast, that is DARPA hard. So again, building capability, clear in your head what you want. You're the government. By the way, we don't do peer review. We do review based upon other government scientists. I use NIH scientists, FDA scientists. I use NSF scientists. Why? Because you have no ulterior motive. The beautiful thing about being at the NCATS is your only desire is the public good. You know, you get paid. I know what you get paid, Jamie. It's very sad. I'm so sorry for that. Your mom probably cries when she hears. But the point is that you guys work super hard, really only for the public good and for the sake of science. There's no alternative motive. You're not in competition with anybody else. So we at DARPA believe those are the kind of people we want reviewing. And are you less expert than other people? Like hell you are. I would argue vociferously that you, the VA, the other people who get derided for not being the professor at Harvard is a lot of bullshit. You guys are as good a scientists as any on the planet. So you should be reviewing these projects. That's what we did. And so and through that, you can have accountability. You can say yes. That's the magic.
Well, thank you so much for that response. We'll have to keep that in mind. And again, I would chime in, say we're the National Center for Advancing Translational Science, not the National Center for Doing Translational Science. And you're going to hear more about that. Not to pat ourselves on the back, but that's going to be a theme when we talk about with Aspire. Sam Michael, you had a comment you want to mention.
I want to say one thing, Jamie. You guys should swagger. You guys need to swagger. No, I'm not joking. I used to have a statement, and a lot of folks know this, that when people would say that is just not doable, or that's not really real science, or blah, blah, blah. Why are you doing it? My answer was a single answer. I said, we're going to do it because we're DARPA. That was only my answer. So what makes you think you can do such a thing? A brain-controlled robot arm. What makes you think you can make a genomic vaccine? They asked Dan Wattendorf. The answer is, we know the science, not possible, not doable, blah, 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 blah. And we look him right in the eye and goes, we're DARPA. That should be your mantra. I know you guys. I was on your advisory council. You're being headed by an amazing scientist by the name of Dr. Rudder. So right now, you guys can do anything. And your answer should be not apology, but walk in the room, swagger in the room, goes, hey, no worries, NCATS is here. You got a problem? NCATS is here. We're here to help you. We're here to advance you. But guess what? Open the doors, open the windows. All things are good because the NCATS is here. That should be your swagger.
Dr. Ling, I really love your passion. I think at NCATS, we do have some of that swagger for sure. And I think you'll see that during Aspire Day. I wanted to comment on what you said about the pricing, because it's something, our experience. I just wanted to, some of the folks are aware of this Patella story is when we started screening for high throughput screening, the cost of a 1536 well plate was about $60 per plate. And now through competition and actual additional sources, we get those depending upon the plate type for around $4. And you'll actually see some techniques where we've actually made that cost even cheaper. So it's not just the fact you're being gouged, but also the fact you have that opportunity cost of the funding can actually be used for some other objective. And so I think that really resonated loudly when you talked about that. Definitely one of the problems that we're trying to solve. And also what you also, I love at NCATS and with Dr. Rudder, we're in great hands, but also the advancing aspect of it, because it's something we also tell our staff is, when do we advance? And the answer is what you just summed up is all the time, right? The advancing is, we are always advancing. We might make small steps. It might not go necessarily straight, but it is a constant evolution, a constant movement. So just wanted to thank you for the talk, but the supply chain we've seen repeatedly that when you have that single source, and even back to Alex's point about the serial process with SPE, those single points of failure become debilitating. So definitely something we try to address at NCATS. But just want to thank you for your passion, because we definitely want to carry that as part of Aspire.
Awesome. I think we have time for one more question, and then Jeff, there may be some questions in the chat that if we don't get to, if you'd be willing just to reach out to those folks. Okay, I guess the last question is, are there any plans with, on-demand pharma to go into like biologic space or non, your classic small molecules, whether it's biologics or more complex, less synthetically accessible, even things like antibiotics?
Yes. So the short answer is yes. Part of the battlefield medicine program, that was the DARPA program, half of it was in fact biologics. And the other half was the small molecules. You guys are all familiar with Chris Love's work up at MIT and also with Govind Rao's work at UMBC. So Govind Rao and I are close friends, and in fact, we're neighbors. But Govind's work is what I like the most for the application that I'm talking about. As a cell-free system, it's amenable to the small footprint that we're talking about. Living systems are problematic in the sense you got to feed the doggone thing. I can't even keep my aquarium at home clean, let alone trying to make medicinals with such a thing. So I like Govind's platform, but Chris has a really good platform. A couple of other folks do as well. Those are all great. The only issue with them, which makes them a little harder to make the easy pivot, and I will make the pivot, by the way, with that platform. I did license out with Govind's blessing and to develop out his platform, which is a cell-free system, to make medicinals. But the problem is you're making biosimilars, not biosames. And so the regulatory burden is actually significantly higher. So for a starting out company, it's much easier to do what I'm doing, and then I'll leverage that to go ahead and then do the biologics. I will say this though, there is one place that this approach is actually helpful in the small molecules. As you know, there are many drugs are semi-synthetic, right? The opioids is a classic example, right? Well, in fact, even the bromocriptine I spoke about is a classic example. The anti-infectives are clearly another example, right? So there is a place where you can actually use these biological cell-free systems to make precursors. And that allows me then to make the final formulated biosame and then not have a less regulatory burden. So we're actually using these systems to help us and as well as mature, so we can mature them at the same time of not assuming a huge regulatory burden at the back end.
Awesome. Thank you so much, Dr. Ling, for this passionate talk. I think we're all, at least at NCATS, we're all fired up. And, you know, from there, I'm going to introduce our next speaker. This is Gergely. He's our informatics lead of Aspire, and he has a background in organic chemistry, biomedical sciences, and chem informatics. He and his team, they're focused on developing the chemical intelligence component of the Aspire platform. And this includes a lot, including synthesis route design, reaction prediction, translation of synthetic routes to reaction templates, all of this that can be executed from an automated platform. And so with that, you know, I'm going to turn it over to Gergely, and he's going to demonstrate or talk about some of our capabilities that we're developing from the informatics side.
Thank you so much, Jamie. So I will share the screen. Do you see my slides?
Yep, perfect. Thank you.
Okay, so thank you so much, Jamie, for the kind introduction and for organizing this Aspire Day. And also I'd like to say thank you for everybody for the opportunity to present our work here and all the work for the work of all the organizers. And I also want to say a huge thanks for Dr. Jeff Ling for a very inspirational talk. And with that said, today I will talk about the recent advancement and innovations of the Aspire Informatics team in the field of reaction informatics. And I would like to mention upfront that this has been a very collaborative teamwork with many teams involved with the Burst IQ Incorporated, Rancho Biosciences, Axon Informatics, and Palantir Technologies. And we are, this research has been a lot of fun, and we are very thankful for these great collaborations. So as Jamie mentioned, the focus of the Aspire Informatics research is the creation of the so-called Aspire Integrated Computational Platform that will provide the chemical intelligence to drive the chemistry automation efforts in Aspire. This integrated platform, in the core of this platform, there is a reaction knowledge base, and it will be equipped with essential modules to do synthesis route design, reaction prediction, condition prediction, and reaction templating, as Alex mentioned, basically the process to turn synthesis routes into protocols that can be executed by the automation platform. We are also building a hypothesis generating engine that actually started because of the COVID pandemic. The idea is that this knowledge base and these modules will act as a molecular designer. So we can design the molecules, the target molecules, we can design the synthesis routes and the procedures to synthesize them. We will store all the protocols and parameters in a database, which will act more as a traditional ELN, and it will hold all the necessary details so that it will be executed by the automation platform. Once the reactions are executed and carried out, then we will collect information so that we can compute the yields and the purity, and then we feed back into our knowledge base so that we can continuously improve the design and the process. And as Alex mentioned, Aspire will operate in a closed loop system. So the molecules that we will synthesize will be tested in biological assays, and we will also feed that information back into a knowledge base that will help with molecular design. Now, when we wanted to when we designed this integrated computational platform and started building it, we needed to bear certain considerations. As you may know, and NCATS is engaged in many collaborative projects that and which are under some collaborative agreements. These are typically bilateral or sometimes multilateral collaborations. So there are some typical questions that arise that represented an open challenge today. For example, what is the technical solution if one of the parties of the collaboration decide that we should bring in a third party into our collaboration and we want to share data with that third party? How can we make sure that that this data sharing happens on a truly consensus basis? Another similar question arises that's very relevant to Aspire, that if chemists want to design synthesis routes, then they would like to utilize the in-house reaction data. But is this something that we should allow, or only chemists should be able to utilize the public domain data when to do the synthesis route planning, or only the data that fall under the collaboration that they are part of, or some combination of these different segments of data? And the similar question arises if somebody wants to build AI ML models utilizing biological and chemistry data. So our idea was to create a technology that can address the secure data storage and data exchange. It provides the means for efficient access control, and also that can keep the flow of the information, basically to keep track of the chain of custody. And so the foundation for synthesis automation comes with the synthesis route design. And since we did not have the technology to address all the challenges that I just mentioned, we couldn't start building our reaction knowledge base from in-house data. So therefore, we needed to use some surrogate data. In this case, we decided to use a subset of the United States Patent Office, the USPTO reaction data set, and we built a reaction knowledge base from 100,000 reactions of this data. And that's what we used to build the prototypes. And also, we are in the progress to integrate the synthetically accessible virtual inventory, the SAVI database that Dr. Mark Niklaus and his team developed at NCI. And with the help of this knowledge base, our aim was to be able to do synthesis route design in a fast, efficient way. And we are also looking for angles like how can we improve the operation at NCATS? So how can the Aspire transform certain practices that we do? And one of the very typical situation in screening campaigns is that once we identify the hit molecules, the lead candidates, then chemists need to resynthesize those compounds, and also they synthesize typically some size of an analog, the SAR series to the lead candidates. This is a task routinely done that we would like to unload the chemists of. So we figured out that in the SAVI database and between the NCATS compound collection, there's an overlap of 130,000 compounds. We also went ahead and found analogs to these compounds in the SAVI database, which at this point is around 13,000 compounds. So with that, we should be able to provide synthetic routes to this number of compounds and their analogs, which, as I said, can routinely done in any screening campaign at NCATS. Now, we also needed to figure out to build an infrastructure and architecture that can support the synthesis route design on a high throughput synthesis setting. And so we needed to find some scalable solutions. And we decided to implement a so-called hybrid database solution, utilizing a graph database, state-of-the-art graph database, the Neo4j database, and the relational database, the PostgreSQL database, equipped with the RDKit cartridge to do cheminformatics operations. In this, the idea here is that we can generate synthesis routes, but we can also store them, so we don't need to recompute them every time for each target molecule. We can store it very efficiently in the graph database, and we can represent them as their INCHI keys. So the idea here is that it also protects chemical information, because with the INCHI keys, it's very hard to figure out what's the underlying structure of the molecule. You can only do that if it's on the public domain. And by also not doing cheminformatics operation in the graph database, we can very easily find synthesis routes, because here we represent reactions as reaction and substance nodes, and the relationship between these nodes tells which substances are the reactant, reagent, and products of a certain reaction. And then we can easily daisy chain these reactions if you're interested to find a synthesis routes for a certain target molecule. And Neo4j can do this very efficiently. We also take advantage of the RxNO reaction ontology. So you have a hierarchy of these ontologies. On the highest level, there's a reaction class. Under it comes the reaction name, and under the reaction name comes the specific reactions in your database. So it's very easy to do queries, for example, what are all the Suzuki reactions in your database. Now, why this is very powerful, but as I mentioned, it doesn't have Cheminformatics capabilities. That's why using the RDKit cartridge, we can perform similarity searches and substructure searches to find reactions that have products or reactants very similar to a query molecule. However, again, for the consideration of protecting chemical or sensitive chemical information, we decided to encrypt the structural information of the molecules. So even if somebody has access to the database, they never can see the raw structure of the compounds. This is something that has not been done at NCATS before. And but that it poses another challenge. But how do we do similarity search and substructure search on an encrypted information? So the solution we came up with was to do the on-the-fly decryption of the structures. So the raw structure is never visible for any user. It only exists in the memory. Why these solutions were required to create a database and architecture that protects chemical information, but it was not complete. We needed to find another solutions to do that, and that is why we turn our attention to the blockchain technology. And as a matter of fact, Dr. Alex Godfrey pointed out this technology to the Aspire team, and Dimitrios Metapsatos found the world-leading burst IQ company in the field of blockchain technology. And we engaged into a pilot project, a collaborative project with the burst IQ company to see how we can adopt their technology to our needs. And for those of you who are not familiar with the blockchain technology, I would like to give a very short and high-level introduction. So in blockchain, you can think of in a blockchain as every record represents one block in the chain. And as you add new records, you chain them together in the blockchain. Now, but the interesting thing is that when you put the record into the first block, then you apply a so-called hashing function on top of the information that this block contains. And that generates this hash value. So that you repeat that process for the next record that you add to the blockchain. But when you generate the hash, now you are feeding the hash value of the first blockchain or the previous blockchain to the next block. So what happens is that all these blocks will be inherently interlinked, and that's why it will create a practically immutable data structure, and also very importantly, data history. And also the blockchain technology has the aspect that whenever a record is accessed, then that event will be recorded, and also like who got access to what information. Another important aspect is the so-called consent contract, because when the blocks are created, they inherit the permission from the owner of who created the record, but sometimes you want to change that ownership. This also provides a scalable solution and requires only minimal manual administration and attention, and provides a very strong encryption for chemical, protecting IP-sensitive chemical information. And we actually created the first-in-class blockchain technology to do that. Here, I'm just showing that in the blockchain, we have this very interesting data structure that we called these data structures ledgers. And we have one ledger for reactions, one for substances. And the message of this slide is basically that on the blockchain, there is no way to reconstitute like what are the constituents of the reaction, what are the reactants, reagents, and products, because there's no association between the reaction IDs and the each key of the substances. The last thing to figure out was how we can make sure that we apply the same access control in every scenario. So the robotic system will be one, so to speak, user, and also there will be the chemists and collaborative partners from even outside NCATS. So typically the requests will come from these sources. So we decided to create an architecture where we have an API, a reaction application, application programming interface. In this case, we call it the RDM as a reaction data management API. And every request will be routed through, or actually is routed through to this API that then delegates and translates these requests to queries and integrates the result. But before sending back to the user, first it will consult with the BurstIQ blockchain API and then with the blockchain itself to see if actually the results that will be returned and knowing who the user is to requesting that information, whether the user should see that result or not. And if the answer is not, or only for a subset of the results is that the user cannot see, then that subset of the results will be reducted, and only then the final set will be returned to the user. And another important aspect, as I mentioned, are the consent contracts. So now we can solve the situation when, let's say, NCATS is in collaboration with one party, an academic party, and let's say the academic partner would like to share data with a third party. So in this case, we can, the academic party can submit the data sharing request to our API, which will be relayed to the NCATS. And if the members of the collaboration consent to this data sharing request, then our API will create a so-called consent contract with the help of the BurstIQ API. And this will change the permission on the blockchain. So now the third party will have access to the data, to the respective set of the data. And all these events are recorded in the blockchain. So it's very important for the terms of reproducibility and transparency. And also I would like to point out that you can think of the third party as actually even like publishing your results. So you can open your results that were protected up to a certain point, but when it comes to publication, you can very easily publish your results very easily with this architecture. And finally, I would like to show you some brief demonstration of these technologies. And we created this user interface that we intend to release as an open source. So everybody can apply it on the top of their in-house database. It was implemented in JavaScript. So it can live as a standalone JavaScript application. But also the idea was, since we use the Palantir platform at NCATS, that we also can use the same interface in the Palantir. And while we were focused on developing this for synthesis route design, but the idea was that we make it generalizable to other scenarios when network analysis and interactive network exploration is necessary, which is actually typically the case when you think about in biological pathway analysis, for example. So the basic function of this interface is, first of all, you can show overlay the structure of the substances on the nodes. Here, the gray node represents a reaction, and you can type in reaction IDs just like in an ELM, and then you can add your reactions to a preview panel to the right. Then you have ways to customize this preview panel, change the grid layout, and you can scroll through your reactions, and then you can swap between what reactions we want to visualize on the main panel. You can even hide this panel to get more real estate on your user interface. But now interesting things that you can do, you can export different data associated with the nodes. For example, you can grab the SMICE of a substance and use it as a target molecule for synthesis route planning. With a click of a button, we execute this scenario, and what you see, this happened real time. So again, I would like to point out we have this reaction knowledge base of 100,000 reactions, and it was that fast to find the reaction routes to this target molecule. Now, you may not think that this synthesis graph is very intuitive, the way it's laid out. In that case, just hang on. In a couple of slides later, I will show you some better representation. Now, I would like to showcase the blockchain in action. So the idea here is that we performed just a simple query when a user will submit the reaction ID. And as you will see, when in this case, my colleague, Viju Matthew, is querying the same reaction ID, he will get a different result as opposed to repeating the same query using another user. And so he's typing in the reaction ID, and then we'll get the result just in a moment. And basically, as you can see, this is even an incomplete reaction. We only have, in this case, it's probably a reagent. So all the other the constituents of the reaction, the product and other reactants, reagents, in this case, my colleague does not have permission on the blockchain to see, therefore they are not shown on the interface. Now the exact same query repeated, just a second, this one. by my colleague, Jayaraman Soundarajan, then you will see that he will see a different set of the substances which are associated with the exact same reaction. And I realized that I need to up my game in video editing, so I couldn't make a nice side-by-side video for you. But in the next slide, I just very quickly, I show the end result. So for the exact same reactions, the two users will see two different results. And that was because of they have different permission on the blockchain. And on the final demo, I would like to show you a very recent development, that how we can lay out the status routes in a much nicer way. So in this case, using a hierarchical layout, it will be very intuitive to see which are the potential synthesis routes to a target molecule. And with that said, I would like to conclude my presentation. So what are the next steps? We will continue the integration of the blockchain functionality into all of our reaction data management API endpoints. We also want to further explore how we can use the BurstIQ platform to enhance the architecture and performance of our knowledge base. We want to extend the synthesis route design features. And also we want to extend the features of the reaction portal to facilitate the collaborative research in Aspire. And with that said, I would like to say thank you for your attention. And again, I'd like to say thank you, all of my colleagues at NCATS. As I said, this has been an excellent teamwork, excellent collaboration with BurstIQ, excellent Informatics, Rancho Biosciences, Palantir Technologies, and also want to say thank to one of our software providers, the Next Move software. Thank you.
Great. Thank you so much, Gary. That was great. And it's, again, I appreciate you putting together this, what we call walkthrough, to try to show people the capabilities that we're building up. And I think this future is very exciting for what we can do in Aspire. I guess I'd open it up if there are any questions that people have, whether it's from the team, Aspire team, or from the audience. Please post them in the chat. We have a few minutes before our next talk.
And maybe, until we get some questions, I just want to again emphasize that using the blockchain technology, we really for the protecting chemical information, but it's the user's choice whether they would like to share the data maybe with the public. So in this case, open data science, but also securing data protection, sensitive information, they go hand in hand. So they're not mutually exclusive. So it's just really a convenient technology. And as I said, it keeps the track of flow of information. So it creates this very transparent like data history.
All right, so we can wait another minute or so, see if there's any. Oh, okay. So we got a great question.
Yes.
About I guess what kind of other data sets are we thinking about pulling in? Whether it's mass spec, other kind of spectroscopy, bioactivities, et cetera. What are, besides this patent data, what are other things that we can think about pulling in?
Yeah, so that's a great question. And analytics data is one of the one that we were very excited to utilize. And again, that's probably an NCAT scenario when we need to navigate the maze. So that data is available. We have that in some data silo, but we need to connect it. And that's what we started to do in Aspire. In fact, Aspire was the first effort to connect the yield data with the ELN data that was not done systematically before. And indeed, using analytics data, we could do systematic reaction knowledge exploration. That's something we are very excited to do. So it's on the next steps, probably towards the Aspire 2.2. That was not the focus of this pilot phase. And of course, biological data, that's something we do to do QSAR and other machine learning and AI projects. Yes.
And we'll be volunteering your team some work in our biology presentation. So there's another question that would you be able to answer in the chat or reach out directly to Dr. Chopra?
Yes, I would do that. Yeah, I will answer in the chat. Yes, thank you.
Okay, so
thank you for the questions.
Yep, thank you so much, Gergely. So we're gonna move right along to our next speaker. So Megav, he graduated from the University of Maryland College Park in 2017, the Master's in Engineering and Robotics, and has been working in NCATS ever since. Megav has been working with biology and chemistry automation, with his work being centered around design and development of low-cost liquid dispensers. With Aspire, Megav is responsible for design, architecture, and development of the DashChem IoT dashboard, along with prototyping benchtop devices like the solid phase extraction that we mentioned earlier and the liquid extraction units. So with that, we're going to turn it over to Megav.
Thank you, Jamie. Hello, everyone, and good afternoon. Let me quickly share my screen, and you can confirm if you're hearing me well and if you're seeing the screen.
Okay, we hear you well. And then I think just hit the present. There we go.
Yep. And I'll actually, in the interest of saving latency, I'll actually stop my video because I have a bunch of demos to show and I don't want them to mess up. So
perfect.
All right. Hello, everyone, and good afternoon, and thank you for joining us here. Today, I'll be talking about DashChem, which is our smart IoT-based lab dashboard, or in other words, what Alex mentioned earlier is our NCATS dashboard. So in his presentation earlier, Alex mentioned that how the way chemistry is performed at the bench hasn't really changed over the years, and how there's not enough influx of automation and adoption of automated systems in chemistry. And that's something that we want to change with the Aspire platform. And DashChem, our NCATS dashboard, is a critical component of it. So with that being said, let's dive right into it. All right. So the first thing that I want to talk about is Internet of Things, or IoT. Now, everyone here must already know about IoT, but just to give a brief description, Internet of Things is basically a collection of devices that are connected with each other through either internet or Wi-Fi or Bluetooth, which allows them to exchange information and data with each other. What this allows us to do as a user is control those devices using an application. This allows us to control them and monitor them. And a prime example of that is home automation systems. Everybody knows about home automation systems, right? We have an application on our phone or tablet, which allows us to, through a use of a sensor, allows us to communicate with physical devices in our houses, like music systems or fans or lights, and allows us to control and monitor those devices. We basically want to replicate that system, that kind of setup, in a lab environment as well. And so what we want to do is create a hardware and software-based infrastructure, which will allow us to integrate practically all the lab devices that are typically used in a chemistry lab and put them up on a single dashboard, a dashboard where a user can log in and access all of those devices and basically interact with them. They should be able to control those devices. They will be able to monitor the devices and monitor the data, monitor the status. So why are we doing this? What are the objectives, and what is the inspiration behind this? So first of all, as I mentioned, we want to take all the devices and create a sort of a homogeneous environment where all the devices are on a single platform and the user can log into that platform and access all of those devices, right? What this allows us to do is everybody can use the same platform to access those devices. And not only that, we also want the user to be able to access those devices remotely. What this does is we have example of COVID in front of us, right? Over the last year, we have seen that the work in lab has suffered a lot because people haven't been able to visit the lab a lot. And even if they have been, there's a lot of restriction in hours. And that basically cuts back the development time. So having this kind of remote access will allow them to basically go into the lab, set up their equipment, set up their reaction, come back to their homes, and practically look at the monitor or look at the dashboard and monitor the reaction. They'll be able to view a live video feed. They'll be able to view the data points that are coming out of the reaction while the reaction is running, all of this in real time. Another thing that we are trying to do is create seamless integrated workflows. So Alex in his presentation also mentioned about integrated workflows, right? That all the devices are connected with each other. Now there's a problem in there that the users have to do a lot of manual labor, that they have to collect and track data manually. And that causes a lot of unnecessary time waste and decreases their efficiency. So that is something that we want to address through the use of this dashboard as well. And I'll talk about these things more in detail as we move forward. So let's talk about the architecture. I have a couple of slides on architecture. I won't go into a lot of detail about it, but tomorrow we have a presentation by Vishaka, and she is going to be talking about the architecture in more detail from an engineering perspective. She'll do a deep dive on the technological details as well. But for today, I'll just show you what it looks like briefly. So in the lab setting, we have a device, and we were working with the H-Cube Pro device, which is a hydrogenation device, and that turned out to be a pilot device. The H-Cube is connected with a Raspberry Pi. Raspberry Pi is basically a microcontroller through which we are running a code. That code sends some commands to the H-Cube, and in turn, the H-Cube returns some data. And that data is basically your telemetry data or reaction data, which is your pressure, temperature, moisture, reagent and solvent volume, and stuff like that. All of that data is then sent to a cloud platform. That cloud platform is basically, for this particular prototype, we used Microsoft's cloud platform, which is the Azure IoT network. The reason why we use this is because we wanted to do quick prototyping and quick iterative work. And that's why Microsoft turned out with the best option for us. And then the job of this cloud network is basically to collect all the data, store it, analyze it, and then finally deliver it. It acts as a mediator between the user who's looking at the data and the devices that are on the premises. And then the cloud network basically presents that data in front of the user in a visual format such that the user will be able to interact with it. Now, this here is a very complex diagram, which is the entire, this is my attempt at encapsulating the entire Aspire platform in one diagram. Obviously, there will be some things missed. And again, I won't go into a lot of detail about it. It's a very complex diagram, and Vishaka will be talking about it more tomorrow. But just to boil it down and just to give a brief essence, I tried to boil down or water down the diagram into its core components. So we have 6 core components that you can see on the bottom left. We have the dashboard, we have the experiment knowledge base and the AICP work that Georgi is doing. We have the chemical inventory work that Alex and Cullen and these guys are doing. We have the instrumentary inventory work, which Carlene and her biology team are doing. And finally, the chemistry activity, which the chemists in the lab are performing. We want to make sure that all these different components that are working individually work together in a cohesive manner, and that we form a closed loop system. And this and the dashboard that we are creating, it remains at the centerpiece of everything, and it sort of brings everything together and ties everything together and helps us create that centerpiece and in a closed loop system. So let's that being said, let's get into the demos and let's get into the inspiration of what started this project. So H-Cube Pro was the first device we started working with, and when we started the Aspire platform, we started talking with chemists and lab managers, and we tried to understand what kind of devices they are using and how much they are using it and what kind of devices are going underutilized. And we gathered a lot of information about these devices. One thing that we got to know specifically about the H-Cube Pro is that it is going quite underutilized. So we tried to investigate what are the problems behind it. So the problem that we found, there are a couple of issues with it. One was a location, that this device was sitting in a dingy corner room, which was essentially converted into a storage room. So nobody wanted to go there. That is now being solved, but there are other issues as well. And the other issues are when a chemist walks up to this device, they don't know what's going on with the device. They don't know whether the device is operational, whether it's broken down, if somebody else is using it, if the device has an upcoming maintenance because of which it is unavailable. And this is not limited just to the H-Cube, but also to other scientific equipment that we use in the lab, right? So we tried to solve this problem. And there's another problem, which is device specific with H-Cube, that the H-Cube needs to run on a daily basis. If it does not, then it dries out. The reagent delivery lines and the solvent delivery lines, they just dry out, and there are air bubbles that form in it, which basically mess up the entire device. So we also needed to make sure that the device is being run on a daily basis. So what we did was we basically connected a Raspberry Pi, which was running a code in which we we started doing these priming reactions, these blank runs, which will basically check each and every functionality of the device on a daily basis. It would do a daily run at like 6 in the morning or something, and then it'll collect data. The data that it collects is basically what happened during that priming run, what kind of reaction parameters were evaluated, what kind of logs were generated. And all of this data would then go to the chemist. The chemist, based on that information, would see would be able to make a better decision that, okay, the device today is active, it's in a running condition, and I can go and schedule my reaction. And that empowers the user to use the device, and it makes sure that the utilities that are provided are not going underutilized. So let's get into the demo of it. So the first thing that I want to show is the status reports, that we're creating status reports on a daily basis by running priming reactions. So here you're seeing the front screen of the dashcam, which has a list of all the devices, right? You have the HQ, BioTouch, and all of that. And you also have these buttons, active and inactive buttons. When a user goes and clicks on that button, they basically see that, okay, the device is operational and it was last primed on this date, on this time. And they can also go and look at the log files of those priming reactions. And this enables the user to basically make a much more informed decision and basically plan out their day or their week or their experiment and so on. And in the same way, if a device is inactive, you basically go and click on the device and it shows that what is the reason behind it. It'll give you an error code, and that error code will again empower the user to use that information to make their future decisions. And this also makes sure that all of the devices that are present in the lab setting are being utilized in a proper way, right? So the success of this POC led us to the next phase of development. So we saw that, okay, now we are able to create a system in which we are able to control the device by running priming reactions on a daily basis. And we are also able to monitor the data that is coming out of those devices, right? So we thought that, okay, let's take it to the next step and let's try to run a real reaction through the interface. So that's what we did next. So our next prototype was basically creating an interface in which we had the user input reaction parameters for an H-cube reaction, right? And they would be able to run a reaction. And while the reaction is running, they would be able to monitor the data coming out of the reaction on their dashboard from a remote location. They'd be able to look at the data. They'd be able to look at the device itself and other useful information. So before I move into the demo, let me show you the slide which talks about that particular screen. So on the top, the yellow boxes you see is basically the experiment info. That experiment info contains the experiment ID, the reaction parameters, and all of those good stuff. On the right side, you see the reaction equation, and I'll talk about this more in detail in my later slides. But this is all relevant information for the user, right? And then on the bottom screen, you see the graphs that are being generated from the data that is being spit out of the device. So while the reaction is running, you're able to visualize the data in real time. On the top right screen, you're seeing a live video feed from the device itself. So we thought that let's provide the user with a live video feed of the device, not just the data itself. So this allows the user to physically look at the device. If they are sitting in a remote location, it allows the user to physically look at the device and make informed decisions on what to do next if there's something that goes wrong. And then on the bottom right screen, we have a log stream, which basically gives you a play-by-play of what's going on, along with like timestamps, and it says that, okay, it's ready, it's connected, all the next steps are happening and stuff like that. So with that, let's get into the demo. So for this particular demo, we basically chose the HQ Pro. For the HQ Pro, we created this infrastructure where the user was asked to input all the various reaction parameters. So along with all these reactant and volume parameters, they're also asked to input some of the system parameters as well. And based on that, the user would run the reaction. Now, once the user started running the reaction, they would be taken to the monitoring screen. And this monitoring screen will basically show you the data that is coming out in real time. On the top right screen, you would see the video feed of the device itself. Here you'll see the data, graph data that is coming out in the log stream. And so basically, while the reaction is running, you can see all the data that is coming out. It's like being in the lab. but at a remote location, and you're able to visualize everything, right? And this camera thing also ended up being its own sort of a side project. Right now, we are collaborating with companies like Verizon and Haivision to create an infrastructure in which we will put cameras in front of most of the devices in the lab, which will allow the users to visually look at the devices if they are not able to be present in the lab for the duration of their experiment. Right. So this was a proof of concept. This was a big success for us, which demonstrated that, okay, we are able to use a dashboard. We are able to remotely log into these devices, and we are able to run a reaction and basically get data off of it. So that led to our next that led to the next step. And the next step was integrating another device. So in this case, we chose the BioTouch V10, which is an evaporator device, which is quite commonly used in the lab setting. So for this particular case, we use the same approach, right? We had the Raspberry Pi, we connected the Raspberry Pi to the V10. We created a control interface, which would allow the user to select the kind of methods they want to run. They would also be allowed to use the number of vials. So this sort of covers the point of that Alex was mentioning about switching, easily switching between single processing or batch processing. So this allows us to do single processing and batch processing as well from the same sort of reaction dashboard. So for this particular demo, again, it's the same thing. We go into the V10, we select the experiment ID. Again, keep this experiment ID in mind. I'll be talking about this in later. But experiment ID is basically a unique identifier for the experiment that you're running, right? And so we come here and we select the kind of method that we want to choose. And then we also select the number of vials that we want to run. And then we just run the evaporation. And here you see on the right hand side, you see the screen, the video feed coming from the BioTouch V10. You can also see that how the V10 is working. And on the left hand side, you can see the pressure graph that is generated by the V10, which also gives you sort of a data point based verification of if things are going well, right? So this was presented and this was done successfully. And yeah, here you see that this is working fine. So this was another successful POC, which basically cemented this idea that now we are able to create a dashboard, which allows us to put these devices on a dashboard, allows us to be in a remote location, and allows us to interact with these devices flawlessly. We are able to control the devices, and we are also able to monitor these devices, right? And this then takes me into, so after integrating two devices, we thought about, okay, we have these individual devices working. Now the next step for us would be to integrate these devices. Now, traditionally, when you see in a lab, the devices that we are using, they are individual devices. They're from individual manufacturers and vendors. So there's no way that those devices would be talking to each other or exchanging information, right? But at the core of IoT, that's what we're trying to change. We are trying to create a scenario in which these devices would actually talk to each other and exchange information. And so this is a wonderful slide that Alex shared in which he talked about how the workflow is integrated on a benchtop and how devices are feeding off of each other. An output of the SPE system will be an input to the evaporation system, and how the output of the evaporation system will be an input to another reaction platform or analytics platform for that matter. So we dove into that world and we tried to figure out what problems people are facing and how can we solve them. So the main problem that we saw was manual labor. So what do I mean by that? So in this particular scene, I'm sharing the slide where it shows that we have an experiment flow where the first step is isolation, second step is evaporation, and third is analytics. It is a fairly straightforward flow, but these workflows can get complex real fast, right? So the problem right now is that if a user, since traditionally all these devices are individual and they're not talking to each other, right? So the user will have to go to the isolation step, and while they're running the step, they have to manually capture the information and track it. Now, when they take that information to the evaporation step, they have to manually input that information from the isolation step into the evaporation step. So there's a lot of manual data tracking and tagging that is required, which is an unnecessary waste of time and reduces the efficiency of the user as well. So we thought that, okay, how can we solve this problem? So the best way to solve this problem would be to create a scenario in which all these devices are talking to each other in a digital fashion. So that's what we did. So since all of these devices are presented on the dashboard and they are in a homogeneous environment, they are all talking to each other. They are all communicating with each other because that is the core essence of IoT. So what we did was, remember I showed you the experiment IDs just now? So the experiment IDs are these unique identifiers which allow you to tag data or track data. So whatever data that is being generated on the isolation step, all of the data is then tracked and tagged, and it is stored along with that experiment ID. Now, when the user goes on to the next step, which is the evaporation step, all they have to do is just click a button and select that experiment ID. And all of that data that was captured in the isolation step now moves on to the evaporation step. And the experiment sort of designs itself by inputting all the reaction parameters. And in the same way, all of the data that is captured in the steps 1 and 2, when you go to the third step, all of that data is again tracked to this experiment ID and then stored or presented as an input to the analysis step. And that sort of takes away a lot of pressure from the chemist to be responsible for tracking the data and tagging the data, and that improves their efficiency by a whole lot. So we were able to do that, and I'll show you a quick demo of it. So right now, the demo we are showing that I'm showing is basically involving all these 3 devices. So I'll perform a reaction on the HQ Pro. Whatever data that is being used for performing this reaction will then be taken onto the BioTouch, and whatever data is being generated on the BioTouch will then be taken onto the SPE. So let's dive into the demo now. So here we are calling this an experiment workflow. So at the beginning, it'll ask you whether you want to register a new experiment ID or select an existing experiment ID. So in this case, we'll just create our own. So remember this experiment ID that I'm creating, it's MV00109, because that is what all the data is being used to tag to. So we create this experiment ID. Right now we go onto the HCube and we set up our reaction. Again, we select the experiment ID. Here you go. And then you set up your reaction. In this, you're basically inputting all the various parameters, your reactant volume, your solvent concentration. All these reaction parameters are very useful. And these are the things that the chemist usually tracks, right? Now, all of the data that is being generated here, you'll see in a minute that it's already automatically tagged. And again, then you move on to the next step and input some system parameters, and then finally start the reaction. And then we come here and select the next device. So for the next device, we'll be selecting the BioTouch V10. And for the V10, when we come in and select the experiment ID, let's go ahead and select the experiment ID. And then you can see that here in the center, we have the experiment metadata tag. And this is just for demo purposes. The UI is obviously always subject matter to change. But here it shows that all the data that was captured in the H-cube reaction is now displayed here in the BioTouch reaction. And it also shows you the current reaction flow, where did all of this data come from, right? And then this basically goes on to show that you're running an experiment on one instrument, you're saving that data to an ID automatically, and then tagging that data and taking on to the next device. And another cool feature for this application is that this application, by the way, you're able to use it on a phone or a tablet, and the phones and tablets have cameras, right? So we created a scanning feature which allows you to basically scan barcodes of any racks or vials or tubes. And you don't need any other device. You can just use your camera, and then basically using your camera, you can scan a barcode, and you can see the number here. And once you scan it, you can submit it, and that is another piece of metadata that is now stored onto this particular ID. Now let's move on to the third step, which is selecting the SPE. And again, in the SPE, we'll go ahead and select the data point or the ID. And again, here you can see that metadata, that whatever metadata we stored, all these 4 points, they are from the H-cube reaction. And then the previous step, in the BioTouch step, we scanned this rack barcode. So we are able to not only scan reaction parameters, but we are also able to scan and tag device consumables or reaction consumables. So it is a homogeneous sort of system in which we are able to incorporate everything. We're able to incorporate the chemistry. We're also able to incorporate the inventory system as well. And again, here on the bottom right, you can see that the current reaction flow, it shows how it's coming from the H-cube and the V10, right? Now, how was all of this possible? All of this was possible because of creating these experiment IDs and then creating a database that allows us to tag all of this data onto that experiment ID, right? So that brings me to my next point, which is integrating the reaction dashboard. Georgi gave an amazing presentation right now and showed how him and his team worked on creating this beautiful reaction dashboard, right, which allows you to visualize structures and chemical structures and whatnot. Now, but the problem is that, well, that's not really a problem, but we want to create a cohesive system. We want to create a closed loop system, right? So for that, so to preserve that essence, we want to make sure that whatever individual developments are being done, they're all controlled and combined together at one location. And that one location is our NCATS dashboard. So whatever work that Georgi and his team have done, they allowed us to, basically by giving us APIs of their website, they allowed us to integrate their work onto our platform. So what that means is we are able to basically access every piece of information that is presented on the reaction dashboard with all the security conditions, of course. So what this allows us to do, and this is just for the demo purposes, what we did was basically take reaction IDs, and based on those reaction IDs, we were able to fetch information regarding that reaction, whether it's inch keys or smiles or UUID. And the next step that we performed was we took those inch keys and we used them to create another function or use another function, which is the Depic molecule function, which allows us to then go ahead and basically get vector graphics of each of the structure in that particular reaction. And using those vector graphics, we are able to reconstruct the reaction equation, which then the user can visualize and the user can understand where everything is coming from and where everything is going. So this demo is going to show you exactly that. So again, here we go. We take the reaction ID, we select the USPTO reaction database, and we select the ID. And then based on that, we get our inch keys. And these inch keys are then presented as an input to the next function, which allows us to get all the vector graphic files. So we'll go on to the next page. And here we'll do get SVG, which is basically the graphic files. And those graphic files will then be reconstructed and presented to you in form of a reaction equation. So here it is. It automatically reconstructs the entire reaction equation, and this is presented to the user onto the dashboard. So whatever work that was done on the reaction dashboard is now integrated and presented onto the NCATS dashboard. And that sort of creates this whole closed loop system, which allows us to perform flawless integrations and allows us to work from one platform to another. And so, and this is just a proof of that. And again, these are just two functions that we have integrated. We will continue to work together and integrate more functions, which will allow us to import more and more functionality. So with that being said, talking about the future work, there's a lot of work that we've done, but as Sam likes to say, the reward of good work is more work. And that's what we are going to do. We are going to do more work, and that work will be in terms of adding more and more devices to the library. Another cool thing that we want to do, and since we have a prototyping capability within NCATS, we want to retrofit existing devices. We want to create hardware solutions which will allow us to retrofit existing devices to create even better automated solutions, which will reduce any extra human work. Another cool thing that we want to do is work with Carlene's group and use the mobile robot and try to completely remove human from the loop and create truly automated experiment workflows. And that's something that we will definitely work on in the future. And then finally, we'd also like to explore any state-of-the-art machine learning or gesture and sound recognition applications, which will basically allow us to integrate more and more technologies into our automated labs. And that being said, I'd like to acknowledge everyone. I would like to thank everyone. Everybody has worked really hard over the last year during COVID, and everybody has performed excellent. And without everyone's help that is mentioned here and a few more other people, none of this would have been possible. So thank you, everyone. Thank you for listening to my talk. And I would like to open the floor to any more questions now.
Yeah, thank you so much for a great demo. It's really exciting to see all the advances we're able to make in spite of a pandemic and everything. So I think with the increased amount of working from home, right, there's a lot of there's a lot of attractive features to this. I guess if there's any questions, please post them in the chat, or I'll open it up to anybody from Spire or, from anywhere across NIH.
Jamie, this is Sam. I have one question. And Meg, a wonderful presentation, and something that Alex has touched on quite a bit. And Carlene will, I think, as well in her talk potentially, or it's like the topic that's come up when it comes to autonomy. What's the take on the reagent delivery, right? Because it's awesome that we can make these things autonomous and we can, potentially automate these. But a lot of this actually does come down to the appropriate materials being where we expect them to be. And Dr. Ling touched on this as well with regards to the production. So, what are next steps you guys are considering with this work for the reagent delivery?
I think that's a good question for Alex as well. I think in terms of reagent delivery, again, just trying to focus more on the in-house prototyping capacities as well, trying to use 3D printing capacities and trying to incorporate those things into the workflow and creating structures based on that. But yeah, I think in terms of reagent delivery, I think Alex and Cullen and these guys would be able to answer in a much better way than I would be.
Yeah, I think when it comes to reagent delivery, I kind of alluded to in my slide is, the bolus of reagents, maybe not every single thing on the shelf, at least in the beginning, needs to fit the bill. But the idea of having these pre-formulated reagents that are accessible robotically, right? And I've always said, it's not that we need one general formulation, right? That beats all other formulations. I'm happy to have dozens of formulations, provided there is actually a robotic solution for delivering that formulation, right? I think that's kind of at least where we want to start with, is like, how many different ways do we want to think about delivering these sets of reagents? Because quite often it'll be dependent on the kind of reaction conditions you're trying to set up that will determine, or the interactivity of the formulating agent, if you will, in the setup of the reaction. And so I think this is a really ripe area for academic research, right? I think this is, if we could tap into our, brain trust in academia to help us understand how do we improve our ability to deliver reagents, I think that would be a really, really important cornerstone to fill.
Yeah, this is it. I have a question also and a comment probably. Megab, that's a great presentation. It's really, you guys have done a fantastic job even doing the thing. In terms of reagent delivery, Alex, I think in the simple, chemistry with those modules and stuff like that, and then some of the things that you talked about, what AbbVie is doing, they may be also advantages to think about some of those. But I have a question. When you are linking up these instruments as they are working remotely and you can control them remotely, if one of them fails or something happens during the middle of the reaction, then what happens? I mean, there has to be a way, somebody has to be there to do that, or are there error correction type of things that you have to do? I was just thinking about that.
This is kind of, you kind of step into this. This is the whole point of sort of iterating quickly, efficiently and so forth, because, this is kind of a systems engineering problem, right? Where you want to build in intrinsic safety devices, right? And understand, you need to intimately understand all the possible failure points. And then even make up some, right? Even, make up some crazy scenarios and have, some kind of, remediation strategy in place. So it's an area where you want to really step in carefully. We've been thinking about this, using video monitoring and so forth. And actually manufacturing industry has a lot of insight around the use of computer vision technologies to actually detect failure modes that are not immediately obvious even to the naked human eye. So those are things that we're definitely thinking about as well.
This also answers the question that Paul actually asked me in DMs. about how do you ensure the system is safe, especially for chemical reactions. So yeah, that what Alex mentioned is spot on, that it's an iterative process, and then we have to take inspiration from the manufacturing industry as well and see how they are doing things and how they are managing to create these assembly lines of creating these products, right? So we can learn from them and can definitely iterate over. And to your point, Sita, when something fails, like we are not at a stage where we can do everything without a human presence. That would be ideal. That is something that can come in the future based on the stuff that based on what we can work with Carlene and the mobile robots and stuff like that. But there'll always have to be some human presence that needs to be there in order to make sure that there's some sort of error recovery or some sort of safety net with a human present there. So
Yeah, the reason I asked that question was we had a similar experience at Lilly in our biology automation lab, and this robot was supposed to load plates for reading into a reader, and something happened and the alignment went away and it started dropping the plates overnight. And when we came out, we found hundreds of plates on the floor. So that was a hard lesson that we learned. I mean, these are some of the things probably it can be addressed much better than what we had to do in those days. We had to have somebody over there making sure the robot was running. That was kind of defeating the purpose. But anyway, thanks. I think great presentation. Thanks a lot.
Thank you. Thank you, guys.
Thank you so much for the talk. I think there might be some questions about mass spec, if you could try to address those in the chat or directly. Just, in the interest of time, we can move along. There's been talk about mobile robots and such. So with that, I'm going to get into our next talk, and let me share my screen. Can everybody see my screen?
Yeah.
So
yes.
Yeah, we're not going to do a introduction of ourselves because that's just awkward. So I'm going to go right into it. So Carlene and I, Carlene is head of automation at NCATS. We're going to talk about the other big part of the Aspire initiative, and that's the biological annotation of the compounds we come up with. And we're going to talk a little bit about some robots because, again, robots are cool. AI, machine learning, optimization, and then a little bit of a teaser about our plans for biological assays that we want to incorporate once we can get back into the lab and go full go now that hopefully the pandemic is receding. So, whoops. All right, so there's a lot of barriers in preclinical discovery, and that's one reason we decided to initiate this Aspire initiative. And just a couple that are relevant for biology. Compounds are made, but we sometimes don't fully appreciate their biological effects. Other times we perform these assays, but when you get the data, it's not in a format that's digestible by the key stakeholders, like medicinal chemists, for example. You generate a terabyte of images, but then what does that mean to the medicinal chemist who's trying to drive SAR? Another thing is, biology experiments can be expensive, right? And again, especially as, a federal program, we want to be responsible with our use of money, right, for the taxpayers. And we all know this, right? Our assays, they're not always relevant to the relevant pathophysiology. A practical problem, turnaround times for biological assays, they can be increased turnaround times, and that reduces the number of iterations. And there's always this issue of just the data sucks, or we can't interpret it right. And so finally, then I think we all have heard about the reproducibility crisis, right? We spend all this money doing these experiments, and then the next person can't reproduce them. So that's a framework for what we're trying to address. And what Aspire Biology, what we want it to be, is these are some buzzwords, but in this talk, we'll talk about what, the substance behind them. We're trying to design this to be adaptable, to allow for more iteration and being on demand, efficient, reproducible. And this is a big effort of mine, is useful, because we can annotate the heck out of any compounds we want, but if it's not going to lead to what's our goal is to help patients, if it's not going to provide useful information, or if it's not even going to be used, it's just going to be dumped on some online repository, then what's the point of doing it, right? And one reason we want to be here with Aspire Day is collaborative. We want input from people, and we'll get to that. And then it's going to be complementary to our existing platforms. So this is not going to replace conventional HTS by any means. We do plenty of that at NCATS, and this is meant to be kind of a supporting or complementary. And we're not going to dedicate this to one particular target, one assay, one biology, and we're going to be able to adapt to whatever we need. So we all know this is your typical, how we think of it is our screening process. We do a screen. Let's see the options. We do a screen. You have to analyze the data. You have to pick the compounds. You do some optimization or retesting. You got to get the sample. You do the experiment, and you iterate over and over again, right? That's how a lot of times this is done in many places, whether it's academics, industry, or government. And what our plan is, and again, lots of people are trying to do this, but we're trying to design a system from the ground up where there's more points of feedback, where we're doing high throughput, whether this is a virtual screen that leads into real compounds being suggested for real biological assays, to analyze it, and then it can feed back into the assays. Or, we select compounds that are potentially of use and to figure out how we can make them, and then just flat out make them, put them back into our libraries and test. And then another component that we're going to talk about with our work with robotics is using this for assay optimization. And so, to give you a sense, once we get back in the lab, what is our module going to consist of? And it's no surprise that we're going to have a lot of the goodies that most high throughput screening labs, conventional ones, are going to have. And because we can leverage the standardization of that's gone with the screening community, we're going to have 96, 384, 15 well formats that are automated, and we're going to have your standard instruments, with the focus on automation. And another thing that we're going to talk about is some of it, we're going to automate things like, our plan is to automate things like the tissue culture. Alex mentioned a little bit about the layout of what this consists of. So we finally got our physical space that we're ready to occupy, and now we get the fun part of putting together the different modules. And so the biology module is going to be flexible, but one component that Carlene's going to touch on in a little bit is, yes, we have this footprint here, and of course I'm telling Alex, Sam, Sita, Joni, we want more, but this is what we get. But we're going to use, our plan is to use technology like autonomous intelligent vehicles, where we're not confined to just this physical space, where we can have an automated TC and have vehicles ferry it. So microplates or whatever vessel we want. Or if something doesn't fit on a multi-mode reader and it needs, let's say, a mass spec, we can take that sample that's prepared with our typical automated systems to specialized readers, whether that's a high content imager, a specialized mass spec or flow cytometry, or again, it's relevant now, what if you're doing something related to a biosafety issue, you can take it to a BSL. I mentioned that one of the core focuses that we're going to have with biology is reproducibility, right? So an example is, what about tissue culture? Errors in tissue culture, you can have misidentification of cell lines, So you have a grad student or a postdoc or even a staff scientist, it happens, right? That's working with 4 different cell lines in the hood at the same time. And they pipette one into the other, and next thing you know, everything's a HeLa cell. Microbial contamination. And a big thing is inconsistent culture practices from person to person. And this can contribute to a lot of imprecision. And so one solution that we have experience with at NCATS is an automated tissue culture system, along with experts in how to support these. And this is going to be one of our plans. And this will be able to support not just one cell line, but multiple kind of core cell lines, and also ones that are specific to projects. And so from here, I'm going to turn over to my colleague, Carlene, to talk about robots and other cool things.
Thank you, Jamie. You've already heard a lot about NCATS plans for Aspire and our expansive goals. You can probably imagine that there will be a ton of consumables involved in the workflows we plan to implement and automate in a continuous and autonomous fashion. We are all more than well aware of that the scientific community's contribution to plastic and other consumable or solvent waste is horrendous. We felt this was another opportunity to leverage our in-depth experience and success with sustainability practices leading to consumable cleaning for reuse, as well as minimizing reagent and solvent waste in our multitude of automated biological activities that will come into play for Aspire. When there's evidence of enough plastic to form a permanent layer in the fossil record, this is not something we can ignore. You can change a slide. Thank you. I'll highlight some of our sustainability tactics to reduce waste. Starting with the video playing on the right, showing our in-house constructed droplet detection system that allows us to monitor dispensing of reagents into plates by a through beam feeding into a custom program that will notify us by text message of any missed wells or leaks according to detected peak discrepancies. This has saved us many times from having to rerun massive amounts of 1536 well microplates. In the middle, we are showing the ion field systems plasma cleaning technology, which essentially vaporizes any organic material on contact. You will hear a snippet on this collaboration in the next talk by Sam Michael, but we have incorporated and integrated multiple ion field cleaning devices into our daily processes, including cleaning of pin tool pins, which are used to deliver nanoliters of compounds to assay plates. The plasma cleaning has led to less rescreening and eliminates DMSO solvent waste. We also use our microplate cleaning system, or MCS, using the same plasma technology to clean microtiter plates used in cell-based assays. Lastly, we are showing one of our systems, which has an integrated Granova pipette tip washer, which we use to clean 384 pipette tip boxes used in functional genomic screening, library, and assay plate preparation. These techniques have been used across multiple processes for years and have shown that the data is equivalent to using a new consumable. Next slide. I love to show this slide because it gives magnitude in a visual sense to the amount of waste we are generating. NCATS is performing many and ever expanding high throughput and automated processes for quantitative HTS and all other peripheral methods that go along with it, such as the never-ending preparation of screening libraries, as well as custom and follow-up plates for confirmation to functional genomics and compound-based screening assays. These processes use massive amount of pipette tips that would normally be tossed into the hazardous waste bins and into the landfill. Next slide. This slide shows the very same process, but now in a modular, continuous, and sustainable manner. We have leveraged use of existing robotic platforms and liquid handlers and have plans to potentially integrate our mobile robot to achieve the continuous loop shown here, but the same goal can just as easily be achieved without it and proper planning. Next slide. Speaking of our mobile robot, speaking of our mobile robot, that is a good segue into what Aspire Biology would gain by incorporating autonomous intelligent vehicles, or AIVs, such as our mobile robot. There are several advantages that this method offers and will increase our capabilities while augmenting existing automation and inherent modularity of some of the planned workflows. Jamie alluded to earlier that there may be scenarios that it makes more sense to make use of a mobile robot or even a drone, which we've explored with another SBIR. It is time consuming and costly to continuously rework large integrated platforms, and in some cases, the need may only be temporary or not on a regular high throughput basis. Some examples could include delivering plates to a high content imager or even picking up dispensed plates with cells from an automated tissue culture robotics system. Next slide. Here is a diagram showing the design of our Biosero mobile robot. It is made up of 3 main components integrated by the scheduling software called Green Button Go. It includes the Omron base, which uses mobile planner software to map the areas of operation in great detail for smooth transport. Omron uses a safety scanner laser as its primary guidance to navigate, comparing the laser readings to a digital map stored in the platform's core. The laser is backed up by rear sonar pairs, a gyroscope, and Hall sensors, plus more. Next, the Cognex vision system is calibrated, making sure this happens at the same height that the nest of the robot will be picking up and dropping off the labware being used to ensure accuracy and proper mobile robot approach and QR code scanning for each position involved in the process. Finally, the precise direct drive arm pick and place locations are taught using the Green Button Go software. The mobile robot is a collaborative AIV, meaning that when a human gets in the way, it will e-stop, wait a few seconds, and recover with no intervention. Next. We wanted to show you a video of the validation we did with the mobile robot literally right before the pandemic. At the time, we didn't have the devices set up or even delivered as we do now. This shows the mobile robot in action, picking up and placing labware at 2 mock locations. The left-hand picture is of a custom and complex system that we designed for the preparation of chemical compounds for various analytical chemistry process needs. And on the right is a high throughput evaporator that samples will be delivered to and from using our mobile robot. As you've seen today, one of the huge challenges we are tackling with Aspire is the automation of chemistry processes. So this was a perfect test case to do proof of concept using an AIV to automate a very manual benchtop protocol. Next slide. This layout shows the plans for integration. There have been a few custom solutions necessary here, including mechanical for loading into the evaporator, since it is a deep nest not easily accessible by traditional robotic grippers, and the benches needed to be lowered in order to accommodate the high nests and platform of the Sirius Omnitasker. We are currently working on the software integration, which will require us to write some custom drivers for these devices, which are not commonly integrated since they are used for chemistry applications as opposed to biology, which has been automated for decades. The API command set used for messaging are very minimal, and we'll have to get creative in this integration. Next slide. You also heard Jamie touch on earlier that we will be leveraging artificial intelligence and machine learning through Aspire workflows, and whether they be synthetic chemistry related, such as reaction screening or biology assay hit selection. If you want to tune in to this topic, will be covered in more depth tomorrow at the poster. Jameson Travers will present at 9:10 a.m. He will show how we used a biochemical assay optimization method as proof of concept, showing we can successfully join the virtual and physical interfaces. The virtual being the robotics machine learning platform in this case, though through the collaboration, but this could also be other platforms like the ones you've heard about being designed in-house that can analyze data in near real time and provide feedback for results-driven iterative experimentation. Next slide. Using the physical system, in this case, the large 4-arm primary screening robotic platform at NCATS, you are limited in how many variables you can test because every time you add another parameter, you add cost, time, and complexity. We ran a brute force experiment with 512 combinations, and it took 5 days, a ton of enzyme and substrate, and generated much waste, except we did wash the plates in this case. We provided for reuse. We proved that we can obtain the same optimal condition for the assay significantly faster in less than 1 day using the AI and ML integration with the robotic system where assays were driven by results in real time. This opens up to near boundaryless continuous and autonomous experimentation when we combine the virtual and physical platforms. Again, you'll hear more tomorrow. Next slide. Now, when we talk about the combined power of what you have so far heard me and Jamie and others talk about, we are in position to shorten the length of time to get through the preclinical portions of the drug development pipeline, which is historically and well known to be labor intensive, takes many years, and prone to failure. Our hope is that with this near real-time iterative and distributive approach, where there's decision-making and feedback throughout, and earlier in the circular process diagram shown several slides back by Jamie, that we will be able to achieve the ultimate goal that is NCATS, get more treatments to all patients faster. The key addition here is the patient and why we do what we do. I'll let Jamie take it from here.
All right, so the plan is we are going to perform both what we call core and project-specific assays. And there's a lot of talk about small molecules and automated chemistry, but are we going to, or will we be capable of testing just small molecules? The answer is no. We plan to include all the goodies, that your targeted protein degraders, DEL, polynucleotides, biologics. The intent is to build up to that and not be restricted to compounds with less than 500 molecular weight. And we're going to have the capability for what we call project-specific assays. And this, again, is going to require adaptability. But again, it may not require a long-term commitment. One thing we can also do that really isn't historically done is we can periodically rerun high-priority targets. This is a great idea from Jim and Glace here at NCATS. So you can imagine as we generate new chemical matter and we have a great automation and everything set up for it, we can go back and retest things, not just one and done. We can also, the plan is to be able to support very specific initiatives, whether that's HEAL, the opioid pandemic, COVID, so-called undruggable or difficult to drug targets, or again, a historic priority of NCATS has been rare and neglected diseases. But the other thing that we can take advantage of this automation is focus on a set of core assays that we would routinely annotate the novel chemical matter that we generate at NCATS or through our partners. And I mentioned earlier, and Dr. Ling mentioned, we're not just doing this for science sake. We want to create useful information or high yield. And this requires a longer term commitment because we have to be very careful about what assays we pick. One thing we're going to do is test everything in multiple concentrations. We do this at QHTS, at NCATS. And again, the dose makes the poison. It's very important that we understand at different concentrations and not just 10 micromolar everything. We can take advantage of things like automated TC and our experience with a variety of high throughput screens to interrogate multiple cell lines or targets in parallel. And we're going to use, our plan is to use orthogonal and also some shared technologies as well. So we don't have to develop 10 different methods for 10 different technologies if we can get the same thing with less technologies, but also provide some orthogonality. We're picking assays that we can link to public databases to maximize their use. And we're going to really focus on using a good set of reference compounds, whether those are bioactive sets or, given my background, also things like nuisance compounds to make sure that we're not going to go after intractable readouts or intractable bioactivity. And we'll also be able to compare, we're going to do genetic perturbations as well. So what are those core assays? These are things like cellular health, physical chemical properties, selectivity, interferences, or things that we want to avoid. And then I'm going to talk just briefly about what I mean by mechanism of action assays. And so one that we plan to really try out is cell painting. And this is originates from Ann Carpenter and others at Broad, where you essentially can perturb your cell line or your tissue with a compound, or it could be siRNA or CRISPR. You can do this in an automated well. You essentially can
There are a lot of storms scheduled for the Maryland area, and I'm not sure if Jamie got knocked out by the storms. We can give it a moment, or maybe what we can propose is if folks have questions or comments from the presentation up to date, Carlene is still available. So maybe what we want to do is take this time for questions while we see if Jamie's going to reconnect. And also great job presenting, Carlene.
Thank you, Sam. Hopefully Jamie will come back. I did see one question about exploring activity-based screening or affinity-based assays rather. And I guess the answer to that is we're not nixing anything that may be useful in this endeavor, and we're open to incorporating all different types of useful data and adding it to allow powerful decision making. I don't know if I saw any. Oh wait, I did. There was something. Okay, and then there was another question, plans to include organoids derived from clinically annotated tissues as part of the screening cascade beyond just cell lines. And that's a great question and well timed, because we do have our 3D tissue biofabrication printing group, and they are working on a multitude of models, including different skin models. They're recently working on an innervated skin model. They have a retina, a lung model in the works, and we definitely plan to incorporate this screening. You know, and they're close to getting into 96 well format, and so those could be some of the things that maybe the mobile robot will be delivering to our platform at some point in time.
Yeah, and actually, Sharad, for that question, when I'm about to present next on collaborations, one I actually touch on in the SBIR portion or specific SBIRs we've done to help enable more biologically relevant tissue models. So we'll definitely expand upon that.
Okay,
Here,
so there's.
sorry about that.
Oh, Jamie, you're back.
Yes. Okay. Sorry about that. So let's
No,
get back. Let's get back to it.
okay.
Loading this up. Oh. So that what I was talking about won't work. So we'll just go right along. You see my screen now?
Yep.
Okay, so the idea, what I was trying to show is that we are going to generate reports that you can interact with this data in a palatable manner. But anyway, just to summarize, our plan is to create a system that's adaptable, high throughput, sustainable, as Carly mentioned, reproducible, but also the most important thing being useful, to focus on our primary mission. And we're going to do this by providing short turnaround times for a set of high yield core assays, but we're also going to support whatever kind of institutional priority we have. And one thing we wanted to take this opportunity with having a captive audience here is if you have a suggested assay you think that would be useful to include, let us know or let me know. And with that, we're happy to take some questions. I can't see them, so if Alex or Sam, if you got.
Yeah, actually, we've got a good one here from Maliga Iyer. Is any thoughts to automate, expedite polypharmacological profiles?
I think that's important because, again, there's a lot of issues with polypharmacology. I think part of that is going to be, I think that's just a big question, right? And part of it is my plan is to really have a really good set of reference compounds, and that can include so-called polypharmacologic compounds, but also the more information we can get about what particular phenotypes or readouts for a well-known probe, that's going to help us dissect some of that. So I'm definitely thinking about that, because again, it's not just one compound, one target.
So Jamie, how about we take up the questions on chat, or at least
Okay.
we can capture them and kind of move on. We're still on schedule, I think, so we may as well give that a shot.
All right. So what I will do, our next talk is going to be by Sam Michael. He's our Chief of Information Technology Research Branch at NCATS. Prior to that, he ran the Automation and Compound Management Group at NCATS, which resulted, they did hundreds of high throughput screens and led to hundreds of probe compounds and several investigational new drugs. He's our federal lead on Aspire. And with that, take it away, Sam.
Jamie, thanks so much. And first of all, thanks everyone for sticking around for the entire Aspire Day and all the prior speakers. Really been awesome. And what I wanted to talk about were actually Aspire collaborations and essentially how NCATS works. And so we're going to reuse some of the slides that you guys have seen before, but just want to really learning the process of repetition.
Yeah, there's supposed to be thunderstorms starting soon.
All right, so someone is on a little bit mute, but sorry. So people have seen, I think it's sorry. So people have seen this slide, and we've talked about what our mission is. And our mission is, innovative methods and technologies to advance the development, testing, and implementation of diagnostics and therapeutics across a wide range of human diseases and conditions. And obviously, Alex showed this slide. Joni talked about this. And we talk a lot about our mission, but then really what I want to talk about is how we get it done. And so also from the NCATS website is this portion, which is what's our approach to actually achieve this. And so, Joni touched on this and you've heard this throughout the day, but we want to talk about the NCATS 3Ds, which are developing new approaches, technologies, demonstrating their usefulness, and then disseminating the data analysis and methodologies to the community. And in particular, collaborations among government, academia, industry, and nonprofit patient organizations are crucial for successful translation. No one organization can succeed alone. And I highlighted that because I want to stress a point. When NCATS is embarking on this mission for Aspire, we are very loudly and clearly saying we cannot build Aspire by ourselves. So to this end, NCATS leads innovative and collaborative approaches in translational science that are cross-cutting and applicable to the broad scientific community. And I put community in bold because, again, you hear at NCATS all the time, collaboration, collaboration, collaboration. What we don't say as much is community. And what we're really trying to establish at NCATS with Aspire is not just the collaborative aspect, because we need to collaborate, we need to build this with the overall scientific community. And that's really what we're trying to establish here. And that's actually part of the reason we were talking about getting an Aspire Day kicked off is we want to start fomenting and fostering and really building this community out, because the scope of this project is massive and we cannot do it alone. And so, Alex showed this slide prior and we talked about the NCATS dashboard, and Megan gave a wonderful presentation, showing this dashboard and the approaches there. Alex talked a lot about the Aspire facility and then Carlene and Jamie just touched on that. And Gergely gave a wonderful presentation about this computational environment, which actually connects the two, right? So this is that virtual physical interface, of how we're going to have these virtual environments to actually initiate experimentation, which Carlene also touched on with the robotics experiment, to run a physical process, be it a chemical synthesis or a biological process. So the scope of this work is absolutely gigantic. And then at the same time, Alex also touched on, as part of this dashboard, is again, how we can actually iterate quickly on different work streams. And one particular work stream here, and the reason I want to highlight this is that we need collaboration at all levels. And so what do I mean by that is we have collaboration at the component level, if you consider this on the consumable side. We need it at the device level for the independent instruments. And then we need it at the system level, which is generating and storing and making use of all of this data. And so I just want to stress, when we talk about collaboration at NCATS, we're talking about big things, little things, everything in between, because the scope of Aspire is gigantic. And so this is, and we want to give some examples. I want to give some examples during this talk of the sorts of collaborations that we've done in the past, and also what we're looking forward to working with the Aspire community. And so this sounds basic, but I want to actually define collaboration, right? And I think this is important. I think Dr. Ling really set the stage for this. So collaboration, very simply, the action of working with someone to produce or create something. And it really sounds simple, but there's so much complexity into this statement. And really the way I look at this, again, is an active process of creation to build capability. And I love that Dr. Ling touched on that when he mentioned, we're not just doing these things for the sake of doing them. We want to build or create a capability that we don't have. And there's a couple things in this sentence again, the action of working, right? So again, this means that there's an active process where people are willing to put in the work. And I absolutely love what he said about saying yes, is if you want to collaborate with NCATS, you have to be willing to say yes, you have to be willing to do work. And I'm not saying this because we don't work with people that want to work. We work with tremendous people that do work all the time. But in many cases, people view collaboration, they mistake it sometimes for a transaction. Rather, I give you this and you give me that. That's a transaction. That's not collaboration. But really, I just wanted to find that going forward. And so much of what Dr. Ling said resonates, especially at NCATS, because you need to be willing to do the work towards creating something. And that's another key piece about this, because I think we all know this, but it's worth repeating. The process of creation is far more difficult than the process of destruction, right? My son, when he was an infant, taught me that, is my baby was unbelievably good at destroying things, right? So when people come to us and they say, hey, we're gonna tear this down, or I can say no, or this isn't gonna work, I say, okay, that's what my infant son could do, right? We hire people and we want to work to make things work, right? That's why we have the expertise we need. And so it's a mentality that we really want to, the partners we look for are interested in making things work. It's great to point out all the failures. We're engineers here, right? So we think of factors, safety, modes of failures, all those things. But the mentality needs to be, what are we going to do to make sure all of these things work? And I just want to stress that point because it's very easy to lose sometimes. So what are the mechanisms to enable collaboration? And this seems a little bit dry, and I'm sorry, this is the dry portion of the talk here, because we have a lot of different mechanisms and we utilize them again. But to the point that Dr. Ling made is we utilize all these mechanisms sometimes in parallel with one another to enhance and increase our capabilities at NCATS. And that's what we're looking to do with Aspire. So we have acquisitions, we have cooperative research and development agreements, we have research collaboration agreements, we have the Small Business Innovative Research grant awards or contract awards, and we have cooperative agreements. I'm going to go into examples of some of these, some specific to Aspire, some to what NCATS has done in the past, again, to sort of show the level of effort and the commitment that it takes sometimes to actually create something. And create something as part of a larger overall community, which is what we're trying to do with Aspire. So first of all, I want to bore everybody with the federal acquisition regulations, because again, language matters and we're part of the government. So there's things we can and cannot do within the government. And luckily the FAR is very, strict, or well, there's room for interpretation, but so I wanted to define some terms. So a contract is a mutually binding legal relationship obligating the seller to furnish the supplies or services and the buyer to pay for them, right? And essentially that this is a transaction in this case. This is what the contract is if we use a contract. Contracting means purchasing, renting, leasing, or otherwise obtaining supplies or services from non-federal sources. Acquisition means the acquiring by contract with appropriated funds of supplies or services by and for the use of the federal government through purchase or lease, whether the supplies or services are already in existence or must be created, developed, demonstrated, and evaluated. And the acquisition process begins at the point when the agency needs are established and includes the description of requirements to satisfy agency needs, solicitation, so on and so forth. The reason I'm defining this is many of the collaborations that we're involved with NCATS, and we'll give specific examples, you saw some today, initiate with an acquisition process. But again, the acquisition of is not the end in itself, it's to increase or potentially create new capability. So I want to give an example of one, which is when we talk about Aspire, and a lot of what you hear is chemical synthesis. Well, the reality is when we first started Aspire, we had no capability to actually automate a chemical synthesis. We didn't have, we have it for biological screening. You've heard, Carlene talk about that. Is we've run, over 1 million 1536 well plates for biological assays. We had zero capability in automating a chemical reaction. And so we worked with a company called Techmatic, and they developed what's called an Indigo mobile reactor. And this is some of the specs. And essentially you saw Alex present on this with the different vials, the different containers, and the different modes that we could run 8 reactions in parallel. And again, just think about that for perspective. We're talking about 1536 well plates to do biological, measurement. We're starting with 8 to actually automate a chemical synthesis. But the reality is this device by itself is worthless. And so I love the example Dr. Ling gave of the stealth fighter, right? Because we have the F-22. As a matter of fact, near and dear to my heart, my first job out of school was working at Northrop Grumman, designing a thermocouple system to measure the temperatures of the F-22 radar. This was in Baltimore. So I'm very familiar with the F-22. The stealth capability isn't just the plane. The plane is one component of an overall system to actually establish air dominance. And this is the very same way. So we can buy the Indigo mobile reactor, but that doesn't give us the capabilities to actually automate chemical synthesis if it's just by itself. We need other components. And so this is where we have different techniques. And in this case, we have a CRADA. So a CRADA is a cooperative research and development agreement. And this is used when NIH and industry collaborate to further develop a technology for commercialization. It doesn't always have to have a goal towards commercialization, but really it's about the relationship with the government and a commercial entity, and we want to develop something together. And so we have the Indigo reactor, and we want to get to a point that we can actually enable automated chemical synthesis. And so we set up a CRADA with a company called Stratios. And so this CRADA was a collaboration between NCATS and Stratios to develop medicinal chemical synthesis workflows using a commercially available automated reactor. That's the acquisition portion, but you notice it's one piece. This collaboration encompassed the delivery of the reactor to be deployed at the Automated Synthetic Chemistry Laboratory operated in Stratios in San Diego, California. NCATS will embed a scientist at the facility to work alongside Stratios personnel while the site acceptance testing for the automated reactor is being performed by the manufacturer. The embedded scientist will also be present as reactions are developed, implemented, and analyzed using the automated reactor, both in a standalone and fully integrated with a larger automated platform capacity. The main goal of this CRADA would be to transfer of our knowledge between NCATS and Stratios to advance the automated medicinal chemistry field. And so I think this is a really fascinating example because we have an acquisition of the Indigo reactor. We have a contract, so we have a contract scientist who is an NCATS contractor working in San Diego. And the value add to Stratios in this case is they didn't have an available spare Indigo reactor that they could actually do offline validation with. We didn't have our lab built yet. As Alex showed, you saw the picture of our 4,400 square foot lab. It's empty, and that just came online a month ago. This was established over 2 years ago at this point. So this is where we have a value add for both parties, but the goal is to advance the automated medicinal chemistry field. And so so this is also a good, time to talk about what makes a good collaborative culture. So first of all, has to be scientific synergy. Secondly, desire and enthusiasm to work together. I cannot stress that enough. Again, everything else becomes details if the desire is truly there to work together. You want to work towards the same goals. We have champions within the parties. We had 2 champions on both the NCATS and the Stratios side. Willingness to communicate frequently. We actually have constant meetings. And then I put this in bold with exclamation, you have to have trust. If you don't have trust in collaborations, you are wasting everyone's time. And we'll get to that point later on. And again, trust is something you cannot put in a contract. I've never seen a contract where there's a portion that says trust. You either have trust or you don't. And what these images are showing here, and you're going to see this tomorrow, this is Mark Ding, who's going to give a talk tomorrow morning at 9:10, which is the development of Indigo reactor for automatically advancing chemical synthesis. And this is actually a trip in March of 2020, before all of the COVID restrictions hit, of Mark showing the first successful automated chemical synthesis using the Indigo reactor. And Mark, again, he can give a talk about this tomorrow. What you see in the bottom right corner, this is Carlene, who just gave that talk, lurking and inspecting the work to make sure it was done properly. But again, it's just us showing the site visit to the San Diego laboratory, and that's the Indigo reactor integrated in its housing so we could actually get this to work. So this is a great example of how we can utilize an acquisition in a CRADA to actually increase our capabilities, right? We didn't have these capabilities at NCATS before, and now we do. And not just through an acquisition, it's through an acquisition, multiple acquisitions, it's through a contract contracts, and it's also through a CRADA, and we can actually achieve something that we couldn't have done by ourselves. Another example of this is we have a CRADA with SciEx, and some of the chatter has been interesting with Tom Covey. I'm really glad that Tom could make it here. Is we have a CRADA, which is essentially, you'll see this talk tomorrow by JJ, which is an open port sampling interface for high throughput mass spectrometry, emergency biotechnology for screening and reaction optimization and drug discovery. And so we had existing capabilities with an acoustic dispenser. We have a SciEx mass spec. This is a 6500 you're seeing here. SciEx also has a technology called this open porous interface, which they actually allowed NCATS to use, so we can incorporate this OPI with our acoustic dispenser, again, to do mass spec screening. This is something that we could not do on ourselves, and it took multiple acquisitions. If you look between buying the SciEx, contracting JJ, buying the EDC, and having this CRADA, we can actually increase our capabilities in ways that we have not done before. Another example that we can use for actually collaboration types are RCAs. And you've actually, with many of the talks you've seen today, you've already seen RCAs in action. And these are used in a collaborative project where confidential information and materials may be exchanging hands. We have many RCAs, and one example, you just saw a wonderful talk from Gergely earlier. For the folks that were on this, we're talking about the collaborative research of use of blockchain technology and translational science. And this was the project with Burst IQ. And you saw Gergely's slide at the end. And what's interesting is this research agreement actually started with an acquisition. We did a microtransaction to Burst IQ to evaluate their technology. Well, that evaluation of technology took some of Burst IQ's time, but you heard all the platforms that Gergely mentioned. We have Palantir, we have contractors with Brancho, we have contractors with Axel Informatics. We actually have our entire contract with Palantir. And so in one small RCA with a microtransaction, again, by itself, I could do a microtransaction with Burst IQ. It's meaningless. It's not increasing our capabilities. It's by the collaborative effort of multiple parties coming together to do work that we actually increase and enhance the capabilities to actually develop a novel technology now that we have a pilot phase and we're thinking about what's next. And so this is one example of an RCA. We have many RCAs. The work that Carlene was talking about with robotics was actually done under an RCA in this case. We had a research collaborative agreement to work closely with robotics, and you'll see that poster tomorrow that Jamie Travers will give. So another technique that we use frequently at NCATS, and Carlene touched on this as well, is the SBIR. I absolutely love the SBIR mechanism. So the Small Business Innovative Research and Small Business Technology Transfer programs are some of the largest source of early stage capital for innovations, innovative small companies in the United States. These programs allow US-owned and operated small businesses to engage in federal research and development that has a strong potential for commercialization. And again, you hear that example is ultimately for some of this, we don't want to just develop this for NCATS. It's great that we get these capabilities in, and you've seen examples that Carlene has shown that we've used SBIRs to bring capabilities in, but the goal is to actually commercialize these so the community as a whole can actually use these technologies. And I want to give some examples of where we've used the SBIRs. Carlene touched on the droplet detection. That was an SBIR. She touched on ion field. We'll go into detail on that. That was an SBIR. And so the whole goal of the SBIR is, twofold. One, to bring technology not just to NCATS, but hopefully a commercial product that other participants can buy. And so an example of this, and someone was asking about tissue models prior, and after the prior session, is in 2019, we submit a contract topic, which was, a lot of people are generating these 3D tissues using transwell plates, but they have a limitation that they use non-degradable membranes. And we've done research at NCATS that have shown that if we use a biodegradable membrane, we actually get better tissue quality and therefore ultimately better science. And so this was a lack of a commercial alternative beyond just the standard, PET membrane surfaces was really holding some of our projects back. And so we thought the SBIR mechanism would be great to hopefully spur someone to have commercialization and provide a solution, not just for NCATS, but also for the community as a whole. And so the goal of this is we said, okay, we want to develop this biodegradable membrane, and these are some of the criteria that we want to make sure it would meet. And the criteria here isn't too important because it's just part of an example. But ideally what we wanted from this was a scalable platform that we can do multiple formats up to about 96 wells. And so we did a phase, this was the clearance form that we had to use. It went to council. And as a matter of fact, Dr. Ling, I remember, asked questions about this. We ended up getting 3 phase 1 awards. And what I want to show next is the outcome of this is this was just the final report delivered by Maytech in April of this year. And what you're looking at is a 96 well biodegradable membrane cell insert plate. And on the bottom, that's actually an H&E stain cross-section of an epiintestinal tissue model that was developed using that plate. And as we speak, NCATS actually has a sleeve of these plates. We have several which are undergoing testing, and Maytech is in the process of turning this into an actual commercial product that they can sell. And I'm touching on this one because you've heard Alex, mention consumables. You've heard Carleen mention, the consumable usage and what we're doing to save. Is again, this is a component level which can have critical impact to the overall system as a whole. And how can we collaborate with parties, within NCATS, but also externally to NCATS to disseminate what we're developing in-house. Another SBIR that we just released in 2020 was a platform for rapidly deployment autonomous laboratory. And I think many folks, are already aware of this as they've heard, during Aspire Day. And the concept proposed here is to develop a next generation of distributed AI-enabled, fully automated laboratories that are capable of functioning in an autonomous fashion and directly linked to a virtual cloud-based resources. Sounds very familiar, right? And really what was pushing this was not just COVID, but the reason we felt this was appropriate for NCATS was we have extensive experience with automation to the component level and have demonstrated the ability to perform autonomous experimentation utilizing a variety of laboratory instrumentation. This concept is greatly aligned with the Aspire initiative, and it would benefit from this SBIR developing modular instrumentation with built-in cloud connectivity. So this also talks about the overall NCATS strategy, which is when it comes to Aspire, we want to utilize every funding mechanism available, one, to build our capabilities in-house, for the intramural program, but two, to actually have financing and potential commercialization opportunities for partners that we work with externally. And so the reason I wanted to also include this example, this was taken from Megav's presentation, this was showing the nominal IoT architecture, because one thing that's also very important to stress is when we talk about SBIRs at NCATS, we like to make sure that we know that what we're proposing can actually be done. So we always take a test case and we do it ourselves. And as a matter of fact, Carleen mentioned the drone SBIR. Megav was actually one of the first interns that we hired to work internally to work on an autonomous drone to actually prove that before we had that SBIR, which is coming to its completion. I'm very excited to show our drones flying materials around. That's coming soon. And so that's how Megav got to start at NCATS, was actually to do a proof of concept for an SBIR topic we want to put out to vendors. So we actually have just gone through the review process of this. There are 2 awardees in the works. We can't disclose who they are yet because it's not public, but this is going to be part of the overall Aspire portfolio, which I'm very excited about. And related to the SBIRs, I think this really, I just want to show an example also of the time that it takes for some of these things. And Carleen alluded to Ion Field, as we actually have a long-standing relationship to Ion Field. And I know this slide is a little bit small, so hopefully folks can see. Maybe I can use the zoom in function if people can see this. I'm going to zoom in here. Is if you see this is obviously we weren't working with Ion Field in 2002. This was when they were first getting started. This is actually, this image, that little purple spark you can see there is the first successful attempt that Paul Hensley, as a physicist, was demonstrated he could generate what he calls as cold plasma to help with the cleaning process. And so I just want to show a timeline of how long it takes for some of these technologies. And I think it was great, Dr. Ling, as well. His slide kind of showed, the idea for ODP happened years before it was actually ready. And so then they had their first cleaning station. They wanted to focus on cleaning tips. They had their first product in 2006. And what's interesting here, and the other reason I want to talk about this in particular with Ion Field, is collaborations change over time. Is their initial product was, we want to clean tips for biological material because they actually couldn't work with DMSO. And now over time, they've actually added the capability, they can actually work with this cleaning process with DMSO. And so this image here that will show from 2009 was showing their first example with a pin tool. If you can see this crust, they went to St. Jude's and they had some, raunchy old pins laying around. And you can see like this crust or whatever this salt is on this pin was not coming off through repeated cleanings in DMSO. one dip in the tip charger and this pin was completely clean. And so somewhere between 2009 and 2013, when we started talking about wanting to clean plates at NCATS, I had reached out as part of an SBIR program, working closely with the Office of Strategic Alliances of putting this on the street. And Ionfield was one of the companies that I saw, well, they're cleaning tips. Could they apply that technology to clean plates? And so they were successful in winning a phase 1 SBIR award. And this is showing a SEM image right here in 2013 of a cleaned well after use. That was good enough for them to get a phase 2 award. And what we're seeing here in this case is a 1536 well plate that was used previously to grow cells, and it was cell titer glow assay washed, and then cells were reseeded and grown. And then you can actually see here that how beautiful this plate looks. This plate has a CV less than 5% with a reused part. But then coupled with that, we also had a need to clean pin tools because we have a 1536 well density. This is the first prototype that they developed for a pin tool cleaning station, which is now in operation on our primary screening system, because even though we're using acoustics, we still have libraries that we pin out of. And the cleaning is so good that the first time Carlene showed this data to the informaticist, you know, the literal response, and this is a scientist, a PhD level scientist, his response was, what is the magic you did to eliminate carryover, right? And the magic was actually a process and a project that initiated all the way back in 2002 that NCATS worked collaboratively with Ionfield in years to develop. And then coupled with that, the SBR phase 2, this one was to develop a plate cleaning system. They actually sold their first unit to AstraZeneca in 2015. And then in parallel, we actually integrated the first commercial version of their pin tool cleaner onto the FDSS, which is a reader from Hamamatsu, similar to the flipper from Molecular Devices. So even though compound addition has been really gone towards acoustic, when you're doing really rapid experimentation like calcium channel assays, you can't acoustically dispense and measure over time. You still have to use a pin. And so in this case, this is a commercial product that we have embedded within the FDSS, and was a huge collaborative work between Ionfield, Hamamatsu, Carlene on the integration, Eric Walgren on the mechanical aspects, myself and the software with the overall dispatcher. Just a project that, again, initiated with a technology first developed in 2002 and now paying dividends that every FDSS assay that runs at NCATS is utilizing this technology. And then finally, you see in 2021, this is actually a picture from the Aspire space that Carlene just took yesterday. This is a commercially available plate cleaning system that we're actually having installed at NCATS, and we're very excited about this. And another thing that Dr. Ling mentioned, you know, when you talk about some of these ideas, they say no, right? And I think this one really rings true is when we first proposed, hey, we want to wash tips, you know, people said, oh, you know, you can just buy as many tips as you need. Try to buy a tip box a day with COVID and see how many tips you can buy without a lead time of 6 months. Same thing when we talked about we want to clean the pin tools, and people said, well, you can just use a DMSO wash because we can buy as much DMSO as you want. Go try to buy DMSO with a lead time and not get price gouged on it right now as we speak. The same thing with plates and consumables. The reason we harp so much on sustainability is not just for the environment's obviously a huge aspect of it, but it's also all the things Dr. Ling was talking about with supply chain, making sure that you actually have access to the components you need to actually perform your experimentation. A true limitation during COVID-19 for actually testing was a limitation in the people being able to buy tips, right? And so now this went from an idea that back when Paul Hensley and NCATS was talking about this, people said, why do you need to do this? You can just buy tips, you can just buy more plates, to now when it comes to the actual microplate cleaning system, they can't build these fast enough because they have so many orders coming in. So this is also part of Aspire, is you have to be willing to be a little bit ahead of your time in some areas. And also there's going to be plenty of people telling you no. I mean, I think we all know that on this phone call. There's a lot of folks saying no. We need to work with people that want to say yes and understand the vision of what we're trying to achieve. And then finally, you know, the other, the final type, and I don't want to steal too much thunder here, is a cooperative agreement, but this is an incredibly important one, is a cooperative agreement is a support mechanism frequently used for high priority research areas that require substantial involvement from NIH program or scientific staff. And I'm stressing this because you'll notice when I mentioned all these different types, what I'm not mentioning are grants. And I'm not saying that we don't like grants. We love grants at NCATS. As a matter of fact, we're the single largest grant program in all of NIH, which is the the CTSA program. But for Aspire to work, we actually need a collaborative agreement with ourselves and the awardee. And so there's actually 2 RFAs which are on the street, and Doda is going to talk about these tomorrow morning with Sean Gardner as well, regarding 2 RFAs, one for physical modules. And so Alex, showed the diagram of the lab and what we're proposing. And the goal here is that we actually want to have new chemistries enabled by the extramural community, have those developed externally, but then bring them to actually integrate on the Aspire platform. So a true marriage of the intramural and extramural programs. And again, NCATS and the Aspire community working together. Another piece of this, which there's also tremendous interest, is we have an RFA for 2 virtual modules, which are all the chem informatics that you've actually heard Gergely and others talk about. Mega, alluded to this, the importance of these. And again, this is something that we need to develop in a virtual environment together. And this is something that NCATS actually has a huge amount of experience in. And just one example of this is, a huge project from NCATS is this National COVID Cohort Collaborative. Even aside from the technical specs of this, which again to date is phenomenal, we have a computational environment with 6.6 billion rows of data, 570 million clinical observations, over 6.2 million patients, and 2 million of those COVID positive. To me, what's far more impressive, this is showing a timeline of when we stood this up from last April. So that's April 2020 to the present day. We actually have 1,403, so over 14, over 1,000 investigators working in our virtual environment. And what's been phenomenal, and the reason the N3C program is working, is what we've established is a community. And so I look at this as a model that we want to establish an Aspire community that people can work together in the environments that NCATS are providing, but we are definitely not building alone. We're building them with the entire community. We're building them with you on the call. Hopefully, if you're, an extramural potential partner, you're a vendor. Again, it comes down to the willingness that folks want to collaborate. And so again, some statistics here, this is, showing the Palantir environment, but this is also, what's interesting when we try to develop are what we call these secure scientific collaborative platforms, and Gergely talked about this as well. So in this case, we're utilizing Palantir to secure patient privacy. In the case Gergely was showing, they're using Palantir and this other technique with Burst IQ to preserve the integrity and potential IP of a chemical structure. So we really try to do things consistently at NCATS the same way, but once we actually do work, we try to actually reuse that work any way we can, because the scope of what we have to work on is immense. But again, the real value of what's made N3C successful is the community, and we would love to foster the same sort of community we would have with that, with the Aspire program. And then another example of this, Jamie was alluding to this regarding the cell painting. We have another great collaborative effort going on, which is a data analysis platform. And this is just another example of an environment that we're working on as a core collaboration for years now between NCATS, between NIST, and between NCI, where the reality is we have a shared development team across 2 organizations, NCATS and NIST, working in a shared code repository to develop an open source data visualization platform that right now is very much focused on high content screening. But the reality is we also want to work with the folks at SciEx and other mass spec vendors. If I can show a high content image of a well, there's no reason I can't bring in a spectra from a mass spec and actually annotate the different chemical components of that. And so these, I know this is a little bit of a whirlwind tour, but I really want to just, manage the expectations and collaborations and just some final talking points. Timeline for agreement, execution, collaboration, initiation. I want to stress this point is counterparts must review our templates, and they have their own layers of review and approvals. These collaborations sometimes take time. We're definitely at NCATS, we're very energetic. We want to work fast, but sometimes getting these collaborations in place, it takes some time, and please be patient. As mentioned before, you cannot draft trust into a contract agreement. Work closely with collaborators and manage the day-to-day issues of the project, set up regular team meetings, make sure scientific delays are communicated early to the entire team. But all of these collaborations that we just shared and the ones you've heard all throughout Aspire have a tremendous layer of trust at the heart of them. And without that, you're just wasting everyone's time. And then finally, again, keep open lines of communication with collaborators and NCATS during the project. If the focus changes, make sure the agreement captures these changes as amendment may be necessary. And again, sometimes these changes are good, and we've actually shown we have many good changes that have led to great discoveries. So finally, I want to say thank you to the entire Aspire team, all of NCATS DPI, in particular, our scientific director, Dr. Anton Semyonov, the Office of Strategic Alliances, NCATS and NIDA Office of Acquisitions. Again, you saw many of these entail an acquisition. Without acquisitions, we can't do any of this. Then the Office of Grants Management and Scientific Review for the SBIR program. And finally, all of our our NCATS collaborators. So thank you so much for the attention.
Hey, thank you, Sam, so much. One question, because we have so many people on before we get to Matt in closing out for the day, is we have a lot of intramural folks. What do you think is the best way if somebody at one of the other NIH sites, or whether it's NCATS or, NIAD or whatever, they have an idea about a possible intramural collaboration? How would you go about contacting Aspire?
I would say, Jamie, you have everyone's emails here at this point. We have and I'm sure the people that, connected on this is reach out to any of the Aspire team members. Because I do think, one thing that's been great about the culture, even within NCATS, is, again, the true number of people specific to Aspire only is not that large. I think it's 8 or 9. But clearly you saw the slide deck that Alex showed with thanking everyone, and there was 30 plus names on there. So I would say if you want to work with Aspire, reach out, shoot someone an email. And we'll communicate that forward. For intramural community, folks outside of NCATS, feel free to reach out to anybody that you've seen present today. I think, most of the emails should be readily available, and just initiate a conversation.
And Sam, maybe you can mention our website, the Aspire website.
That's right. Thank you,
Yep.
Gergely. And we also have the NCATS Aspire website. In many cases, that a lot of that's routing to Dota. You'll hear her talk tomorrow. Dota is absolutely fantastic. We couldn't do this without her, but if people have an inquiry, Dota makes sure she routes it to the appropriate people. So thank you, Gergely. I think a lot of work has gone, a really lot of work has gone into this case with making sure that we have the points of contacts established.
Great. Thank you so much, Sammy. I want to reiterate to all the intramural folks that we're very approachable here at NCATS. If you have some idea or way that you potentially want to collaborate, please let us know. And we'll follow up, because again, we really, our mission is to help patients, and so we're not specific to one set of diseases or one pathophysiology. So again, please reach out. In the interest of time, I'm going to move on now to introduce Dr. Matt Hall, who's going to close out for the day. He's our director of the early translation branch in the Department of Preclinical Innovation here at NCATS. He joined us in 2015. Prior to this, he did his PhD at the University of Sydney. He postdoced at NCI. He's done extensive work in multi-drug resistance, including a lot of work on cisplatin. I should mention if he is active on Twitter, so he's at CISPT2. So if you ever want to connect, I should mention that. So Matt, take it away.
Thanks, Jamie. You can hear me okay? Wonderful. Yeah, thank you so much. So in the interest of time, I don't want to keep everyone here forever, so I'll just make a few brief comments. But today was really about hearing about a lot of the Aspire program and what the NCATS vision is generally, and how we're trying to bring the way that we work to advancing challenges around both the practicing of chemistry and automating it, and also integrating that with the digitization and the informatics of chemistry, and then bringing that into a closed loop with biology. And the lab you saw had everything connected up, and you can think of that the way we always talk about humans, us as scientists and humans collaborating and connecting, and this lab is really the physical manifestation of that with equipment. Obviously takes a lot of thought to achieve this, and Sam finished off there by talking about collaborations, and truly collaboration is the center of everything, and you can imagine there are many versions of that. And so tomorrow is really going to be about conversations. Dr. Austin, our immediate past director, who really instigated at the establishment of Aspire, is going to open the day. And there's going to be some breakout sessions, virtual poster sessions following that, where we are going to have people talking. If you're a biologist, there'll be a biology room for you. There'll be one for chemistry, one for informatics, and critically one for extramural. And a lot of what Sam talked about this afternoon was really focused on collaboration with private companies and invention. Obviously, there are many paths to success within this field, and I know for the academic chemists that are listening or will listen to the recording, using a closed system like the one that NCATS is building to advance the science of chemistry and the practice of chemistry for drug development is a really critical part of all this. And there are multiple dimensions and multiple versions of collaboration. Sam touched on the fact that instruments that people develop either in collaboration with NCATS or through these extramural programs directly can literally be brought on board, and the Aspire platform will be a test bed for really assessing how these perform, how they can be integrated, pressure testing them, finding their strengths and their weaknesses. And then the conversation will continue. There's 2 roundtable discussions. One really talking about how to make the most of a collaboration and what constitutes a successful collaboration. Alex Godfrey's going to lead that, who you heard speak earlier today. And there'll be a second roundtable discussion that really gets to the core of, what will a medicinal chemist be in 5 or 10 years time? How do we train medicinal chemists for that lab? And, hopefully we plumb the depths of the psychological issues around automating chemistry. Biology's been automated forever, and all it means is that our chemists work towards automating their our biologists work towards automating their assays and integrating them and working with automation engineers. The same future awaits for chemists, and we're seeing it already. It's not about replacing chemists with robots, it's about training chemists to work with automation. And I'm really looking forward to that conversation. And again, how we integrate that with things like AI or machine learning on the route mapping and the route planning side, as well as chem informaticians and structural biologists when you get into drug design and actually the molecules you need that you want to dial up. I'm really looking forward to that conversation as well, because I think we really need to be thinking now about the chemistry labs we build. Do they need room for a robot to move through the room? Do they need flat open benches? Do they need different types of hoods to accommodate the infrastructure of, well, literally today and tomorrow, so that will be very normal and standard. So I'm looking forward to tomorrow, and I really hope that you all come back and join us, understand more about how NCATS is thinking about the automation and integration of chemistry and chemical techniques into drug development, into translational science as we try to advance it. And hopefully I think of opportunities to work with us. And as I say, there are many dimensions to what those collaborations can look like. So Jamie, I hand back to you, and I look forward to seeing everyone in the morning.
Great, thank you so much, Matt. And yeah, I think being, within 5 minutes of the itinerary, I think we did a good job. And so I think we've all been on the other end, right, where this goes way too long. So again, I think thank you so much for everybody. I'll send out an email later tonight or early in the morning, just a reminder about the link for tomorrow. It's a different Zoom link, a second one, and I'll send those reminders. Again, thank you for your participation today, and I look forward to continuing this discussion tomorrow.
Have a good night, everyone.
Thanks, Jamie. Thanks, everyone.
Thank you, guys.
Thanks, everyone.
Thank you. Bye-bye.
Thanks, everyone. Bye.
Thanks.
Thanks, bye.